Good morning, everyone. Welcome to Swalife Biotech 4th Annual International Conference 2026, Day 2. I, Ruchira Pawar, a warm welcome to all to Day 2 of Swalife Biotech 4th Annual International Conference. We hope you had an experience during yesterday's sessions. Today's promise another exciting scientific journey with expert keynote lectures, outstanding oral presentation, the prestigious award ceremony, and recognition of excellence in research. On behalf of entire organization committee, I extend my heartfelt gratitude to every participant for being a part of this conference. Uh, Without any further delay, let us begin. It is my privilege to introduce our keynote speaker for the first session of day 2, Dr. Deepak Behra sir. Dr. Deepak Behra sir is an accomplished cetacean researcher and academician currently serving as an assistant professor with extensive expertise in advanced statistical modeling, data science, artificial intelligence, and interdisciplinary research. With scientific contribution to research, publication, patents, academic collaboration, and AI-enabled decision-making, he has actively promoted the integration of statistical methodologies and emerging technologies across research and higher education. His work continues to inspire researchers, educators, and students to harness data driven approaches for innovation and scientific excellence. Uh, Today, Dr. Behra sir will deliver keynote lecture on generative AI in healthcare. Artificial intelligence is rapidly transforming healthcare through intelligence automation, clinical decision support, medical imaging, research acceleration, and personalized patient care. We are honored to have Dr. Behra sir share his valuable expertise with us. Everyone, please join me in welcoming Dr. Behra sir with big round of applause. Sir, the stage is all yours.
Thank you very much, ma'am, for the introduction and great opportunity. May I start?
Yes, sir, you can start now.
Okay. Thank you very much, ma'am. Um, Good morning, everyone, and respected dignitaries, distinguished guests, and fellow researchers, academicians, and the healthcare professionals, all the participants. It is my great pleasure and privilege to be with you today at the fourth annual International Conference 2026. I am very happy to have this opportunity to deliver as a keynote speaker. Today, I will going to talk about generative AI in healthcare, particularly in the topic of that means three aspects, important aspects, that is patient care and medical research and clinical decision making. How you have to make the that means patient care, take care about the patients, and how we are doing the research in medical field, and how you have to take the decision in the field of medical and research, healthcare research. Before I will talk about artificial intelligence, I will begin with what do you mean by artificial intelligence, how we are doing the artificial intelligence, the perspective about explore with the today. Today, let's begin about how we are using the artificial intelligence and generative AI in the healthcare sector for betterment, that means advanced uses. Let me explain about how we are going to divide. Going to talk about here. I will talk about these concepts. That means how we are going to talk about, that is patient care, taking care about patients and advancing uses of medical research. So these topics I will discuss about these things. Then why is generative AI in this? That means nowadays we are talking about the generative AI. What do you mean by that, and what is the necessity of that? Before that, first, healthcare is a genetic and enormous amount of digital information. That means, with respect of health, it is very much important for us, and our life is very much valuable. So we have an electronic health records, to maintain the records of individual health cares or individual health personals. So before that we have to that means it is very much necessary to maintain our records, what is the health problems, how to diagnose that. So we have to take care of that, and it is most necessary to maintain an electronic manner. So we are seeing the converges three things, that is the data, or how to computing that data, or how to use the data in the future concepts. So before that I have to discuss about that means generative AI. What do you mean by generative AI? AI can read the work with language. That means it is you can say the language is that means this model developed by with using large language models. That means that can summarize the clinical notes or clinical records, what are the diagnosis already happened or going to be happened. That means AI can create the models, different models, generative models can create by using your data, different different diagnosis data or your medical reports. So it can be helpful for your diagnosis and for health concepts. Uh, in another concepts in healthcare, it is really interesting that part of these abilities can connect it to the real clinical and research workflows. That means by using this generative AI we can do the research in the medical field, and that means you can do the research in medical field and healthcare sectors. So first of all, you have to familiar about the that means how we are using the generative AI and what are the families of generative AI. There are four families of generative AI. These are that means large language model and diffusion models, then GANs, then multinodal and foundation models. So these are the four models we will discuss, and that means basically we are using for AI large language models. And basically we are using the large language models, and recently that means the most in present case we are using the multinodal or foundational models. So the evolution of AI in healthcare has been using forms of artificial intelligence for the decades. In early period, we had rule-based systems that is described by capture expert knowledge. That means the professionals or the experts saying that this knowledge is good. You have to follow these things. You have to follow this. Nowadays, professional is required, but if you are integrating the AI with respect to that, then it will be benefited, and very short period of time we will understand that how what are the problems, how to understand that. So it is most necessary to integrate the generative AI or artificial intelligence in our healthcare sector for the professionals or for the research also in the medical fields. So there are three pillars we will discuss about in this topic. First is patient care, and second one is medical research, and third one is clinical decision making. That means here we will take about care, research, and decision. Care means here we will talk about patient care, how you have to take the patient care of patients, and for second one is research, why and how the research are going on using the generative AI in the medical fields or healthcare sector. Then third one is clinical decision making. How we are take the decision, how we are taking take the decision, intelligent decision using that generative AI. That must be better decision, or you have to for the professional use you have to integrate the generative AI or artificial intelligence in our medical fields or healthcare sectors, so that we will get the better decision. So now we will discuss about patient care. That means how the transferring patient care is going on. That means using the generative AI, how it is going on, or how it is formed when integration of AI is going on. Let us now move to the that means technology is now it is a very valuable, and only when it improves the experience. It is very much valuable for the you can say safety and access efficiency. Technology collect all the data when we are giving some data or intelligent integrated AI with our systems in the medical fields or in healthcare sector. It takes your data and that means increase their efficiency to or increase their critical ability to think deeper. So in this field, that means AI can reduce the unnecessary workload and help the healthcare professional spend more meaningful time with the patients. That means because nowadays it is important to work. Time is necessary, money, manpower is necessary, and cost is necessary. So to reduce that, we can integrate the artificial intelligence with our medical fields and healthcare sector to reduce all these things because AI can work 24 hour into 7. So AI can understand all the data, all the information, but with respect to we have to verify it with respect to their using the professionals. That means manpower is necessary, but if AI is integrated, that the workload is that means easier and it is efficient. We can do the in efficient manner, and AI can understand what are the information or what are the diagnosis is already going on. We have to give the inputs only. Then the that means here we have to give the documents or data to the AI for their analysis or what are the diagnosis going on. We have to go give the inputs, different inputs, so that they can understand what I have to do or what they have to do further. So after that, a professional can verify all these things, what is going on, all like this. So the and ambient that means clinical documentation and AI scribes. That means the important point is that AI is not becoming the doctor. AI become not be a doctor, but he can suggest with the professional. That means when it is integrated, then the professional will be very efficient. Uh, they can do the work in a efficient manner, or it is very easy to do the work. So the AI prepares a draft, that means the treating clinical reviews it and corrects if needed, and signs it before it becomes part of the medical record. So the principle is very simple. AI generates the clinical verifies. That means with respect to integration with the professionals or with integration of the systems, AI can understand all these things, or AI can suggest or do the things automatically. So these are the personalized treatment plans. Before that, we have to give the records and give the data to the when integration of AI is done. We have to give the inputs and give the data. What are the treatments and what are the plans, so that AI can understand all these things and do the things accordingly. So like this we have to use the professionals or doctors to learn all these things and how to put the data so that AI can understand all these things and do the things accordingly. Then virtual health assistant and chatbots, which is very much necessary in this that means in busy this lifestyle busy lifestyles. It is very much understand necessary how to understand the health our health. So that means is there any problem or not, how to understand that. And uh, virtual health assistant means it is a, you can say it is your friend, so that in 24 into 7, when is your some problems like this, you can assist or you can take help from that, because that data or information already integrated with the professionals, that means that are verified, so that the health assistant or chatbots gives the verified information only, because that was already integrated and professional given the data, and it is already that means uploaded in that software or that application. So that any person asking there about queries, some health problems, that virtual assistant or chatbots can help them. how we are going on, what is the problems, for this problem how I have to take care of this. So like this virtual assistant, like a friend, they can suggest you what to do, what not to do. So like this, it is a friend, you can say. So it is available in 24 into 7, and the information is verified by the professionals already. So nowadays it is very much necessary, because in nowadays different things are going on, different problems, different challenges, new diseases are going on, different virus. So these things are necessary. That means it is already updating with the professionals, and verification already going on in the background. So it is very much important to develop the virtual health assistant or chatbots with the integration of AI and the professionals. It is verified with the professionals. So it is very much important to each one of us. Then how to, that means monitoring, and that means remote monitoring. That means every person have that means different problems, because no, that means there are so many patients, so many patients, but bed is so less. So you have to take the rest in our home or like that. So how to monitor the patients? So it is most necessary that you have to develop a software, or develop a AI system with integration of the professional, so that professional can remotely monitoring them, and AI can monitoring them as well. That means when professionals are not available, so AI can able to monitor them remotely. If some problem is arises, they can inform the professionals, or what is going on in the background. So mental health and behavioral support from support from AI. That means it is very much important nowadays. Nowadays, different health, mental health problems, or disorders, you can say, or behavioral changes is going on in our lifestyle or day-to-day life. So to look after these things, that means mental health and behavioral support, we have to integrate AI with respect to verification from the professionals. That means it is integrated with the medical healthcare sector and medical fields. That means how the mental health is changing, how the behavioral is changing, what are the things is going on, so they can send their problems, so that AI can give the answers automatically, because it is the information already verified by the professionals. So it is already created or integrated with the professionals. So it is nowadays a mental health problem already going on, because lot of work pressure or like that. That means so much stress, pressure, like that. So different problems are going on. So to look after this mental health behavioral support, that's the integration of this AI for learning, or for giving this information. AI integration of AI is most necessary with the professionals also. Patient care in action. So let us now look at a couple practical exams. Some examples are, that means those slides highlights a ambient AI documentation, as you see in the slide. So there are 150 health systems. It is also presents, that means the outcomes reported from the health system, including decrease after hours, or chatting, that means increase in direct face-to-face time with patients. Then what is interesting about these examples is not only the technology. The more important is question is, what happens to the clinicians time after the technology is introduced. If the technology is introduced, then the clinician or professionals, what they will do? If the technology is introduced, or technology will do everything, then what is the use of that means professionals? Because if AI can reduce repetitive documentation, that means if we do the documentation, AI can reduce these things, repetitive documentation, and the time saved can potentially be returned to the patient. That means doctors or professionals doing the some documentation, different documentation things, so it can be reduced by when we are integrating the AI system with our healthcare system. So it is very much important to, that means, decrease these things, because doctor can or professional can give more time to the patients or care about the patients. That's why technology becomes meaningful in the healthcare. AI cannot take the place of professionals, but AI can reduces your work or some repetition works like documentation, some documentation. So AI cannot replace the professionals. Professional can give more time to the patients, because they can their AI is introduced, so that they cannot be they can that documentation is not necessary every time, because documentation done by the AI when AI is integrated. So we have to look forward it. And what is the use of medical research when AI is introduced, or how using the AI we can do or advance in the medical research. Researchers have to understand the literature and identify the biological mechanism, discover possible drug candidates, and design the experiments, recruit patient, and analyze evidence. The generative AI can support several of things at the different stages. The important idea is not that AI magically turns years research into few clicks. That means AI can help researchers ask better questions, explore more hypotheses, move promising ideas forward more efficiently. That means AI can help how you have to select the, or how you have to collect the data, how you have to analyze the data. That means it can be that means it can help you how to do the things. We cannot replace, but AI can help you how to do the things. You must understand that. Then drug discovery, how we are using the AI for the drug discovery. Drug discovery, actually it is a very long and complex process, but researchers may need to explore a very large number of possible molecules before finding candidates that are worth testing. That means generative models can propose new candidate molecules or help to predict properties such as binding and toxicity. That means this allows researchers to prioritize candidates before investigating heavily in the laboratory work. So the most important message is that AI does not eliminate the laboratory, but it helps the researchers to decide where the laboratory effort may be most valuable. So the AI can help us search the enormous space of possibility more intellectually, or we can take the decision more intellectually. Then genomics and precision medicine research, how we are doing all these things. That means the generative AI models can help researchers interpret the variants and study possible effects on protein function. As we know, it can be combined different type of information, genomic, clinical, and imaging data. Uh, so this is often described that multiomics and integrated approach. So our aim is to better understand which patient may respond to which therapies. That means, with respect to with integration of AI, we can understand that which patient is that means respond to the which therapy, or which therapy is applicable for the individual patients. So AI may help us to make better use of information we already have, or explore the patterns that are difficult to identify manually, because we are in medical fields, different patients have different therapies going on, or different diagnosis going on. So all this data you have to upload to the AI or in the electronical manner, so that AI can understand all these things and create a pattern. When we are doing the research or when we are doing the diagnosis for the another patient, AI can understand for this patient, or if some symptoms is this, then AI can suggest you for this patient this therapy is applicable. so that we can it will be easy for us and it is benefited for all the professionals. Then synthetic patient data. So that means this is a idea to generate artificial records and reproduce useful statistical patterns without directly exposing real individual patients data or records. That means you have to input all this data to the to in electronic manner so that it can generate the patient data. However, synthetic does not mean automatically correct. Researchers still need to check whether the generated data realistically represents the characteristics uh, data that matters the research question. That means researchers must verify and check the data which was generated by the artificial intelligence or generative AI. So synthetic data should be treated as a research tool that requires validation. So you have to validate the data is correct or not, because not as an perfect substitute for the real world evidence. Because we have to validate the data is generated by the AI is correct or not. So researchers must validate the data and verify the data. Then literature synthesis and evidence discover. As researchers, we all know that one major problem is there are simply too much literature, different literatures available. So thousands of papers can be published around a research area, and it is very difficult to read everything. But generative AI can help you for the summarizing and cross-link large bodies of literature. That means there are different literature are available, but for us or for researchers it is impossible to linkage all these things. But if you gives commands or upload your data to the AI, it can be summarize all the things and cross link between the all the research and literatures. So this can make literature reviews systematic preparation and grant the writing and protocol development more efficient manner. But there is one rule I strongly recommended: never treat an AI generated literature summary as the final evidence. You have to, before that, you have to go through that and check all the things, all the original things are available or not. So you have to recheck all the things, the methods are correct or not, wrong, or the results what are generated by the AI is correct or wrong. So all these things you have to verify and check that before you have to do something or proceed for that. So AI can only accelerate the things or research process, but critical thinking remains the responsibility of the researcher. Only AI can do all these things. That means you can accelerate or speed of your research work, but you have to take care all the things. That means you have to verify all the things, all the data all correct or wrong. Then clinical trial design and patient matching. You have to think it is very much important. That means how you are doing the clinical trial design. So one of major challenges is finding patients who actually meet the eligibility criteria for a clinical trial. Our important information may be present in a unstructured clinical notes rather than neatly organized fields. But when we are doing those things, we are unable to structure all the things. So we are noted in a unstructured manner. So AI can help and read all the things, all the notes, and identify the potential trial matches. And it can that means accelerate your work potential manner. So it can also support simulation and trial designs and endpoints before the study begins. That means that can help the research teams identify the possible statistical and design problems earlier. For that means for opportunity is representation. That means to design the carefully, AI can help the researchers identify and recruit more diverse patient population, so that it will be very precise and give the result in precise manner. It is also better matching, that means better planning, potentially more representative in research. So you have to take care of, or when you are doing giving the unstructured documents or data to the AI can give you the structured data or in the which is necessary, and how you have to do the clinical trials and how you have to design and develop that clinical trials. You have to give the command for that. Then research in action. So AI can increasingly being used for difficult scientific problems involving in biological structures, molecules, and drug development. It is AI can use in every sector. So, but we should keep in mind that the scientific principle must be clear. That means AI can generate only hypothesis is beginning of validation, not end of validation. That means from starting, AI can check that which one we have to take the validate. That means hypothesis, but we have to validate that hypothesis is correct or wrong. That means researcher must take care of that. So, in the laboratory and clinical research, statistical analysis and scientific review remain the essential. The researcher must take care of all these things, but AI can help us how to decide all these things. Then powering clinical decision making, how we are take the decisions with respect to in healthcare sector. Now we come to the third pillar, that is clinical decision making. This is AI can support the doctors. Our question is, AI can support the doctors? Yes, AI can support the doctors, but can it identify the patterns, summarizing all these things? That means yes, AI can identify your patterns. What are the diagnosis going on? What are the therapies going on? All these patterns AI can understand and keep that what to do the further. But it should be keep in mind that professionals and researchers must be there to validate that and to verify that. Uh, my AI should argument that clinical judgment not replace clinical. That means AI cannot replace the doctors, but AI can accelerate the doctor's work or doctors. That means potentials. Then medical imaging and radiology AI. That means here we are discussing about when medical imaging is the one of the leading areas of the healthcare sector. When we are integrating all these things with respect to AI and verify with the professionals, then AI can support different concepts like radiologists in several manners, or it can draft the reports from the scans. That means when we are scanning something or diagnosing something, they can create the reports using the AI, or using this. That means when we are integrating AI with the different tools or different machines, so they can create the professional drafts, and it must be verified and checked by the professionals. But the more useful model is AI helps analyze and prioritize, but the radiologist must review and interpret all these things. AI can help only specialists focus attention where the different matters or manual things must be checked and verified by the professionals. Pathology, dermatology, and pathology AI. That means when we are integrating all these things with the AI, you can say in digital pathology, AI can pre-scan large slides and highlight suspicious areas for the pathologist. But in the case of dermatologist, the image-based tools can help prioritize concerning lesions and specialized review. So our common theme is, that means it is most important to that AI not necessarily doing the whole work, because AI can only read all the things what is input there or what is there in the database. Instead of that, it can help the specialist to manage the large amount of information and focus their expert attention where it is most needed. So the professional must take care of all these things, because AI can do only how to. That means whatever the data available, they can only showcase all these things, or they can generate the reports. They can summarize all these things, but professional must verify and give the suggestions what to do and how they have to take care of that. Then decision support, how we are taking the decisions in healthcare sector. When we are talking about clinical decision support, it becomes more useful when it appears at the moment of a decision is being made. For example, if we'll talk about a differential diagnosis assist may suggest a possibility that clinician has not immediately considered. That means when we are going through different diagnoses, maybe clinics are not available or professionals not available, so we are asking about the AI what are the things or how to what are the reports in this. We have to take the support of the AI, but AI should not simply be powerful, because you must take the that means consultation of the professionals. This is correct or wrong, you must be verified before that. Then predictive risk models in the practice. The figure on this slide that reports substantial use of the predictive AI in acute care of hospitals. That means here actually high reported performance in a heart attack, that is rule out workflow. Uh, Here we have AI adaptation among neurologists and more than 1200 FDA authorized and AI enabled medical devices in active use. So the key idea is not the individual number. The key idea is the direction how to travel, and the how the predictive system is being used. How that means flag deterring the patients, prioritize the high risk cases, and route alerts to the appropriate clinician. Our goal is to identify the risk before a crisis, and rather than after the crisis has already happened. But when we are doing the prediction, should never be confused with the certainty. So you must take care of all these things. That means a risk alert is signal for the professional attention. That means we must. That means the professional must take care of all these things, and they must take attention of when we are using the AI. But in the case of that means when we are talking about clinical decision making in the action, when we are taking the actions, that means in the emergency care, the reliable workflow can provide useful information faster. It may help clinical teams make decisions more efficiently. And in the second case, neurology, where the slides reports high adaptation of AI tools. This example show that clinical AI is moving toward everyday practice. That means when we are using, that means practicing day by day, then AI can learn better, or they are very efficiently, they can take the decision very perfectly. So but by adapting should never be only a goal, but we should need appropriate validation or clinical training, monitoring, and clear responsibility by the professionals. You must take care of that. Then in India and global landscape, how the that means AI is using in the healthcare sector. India has a large and diverse population, a growing technology ecosystem, major healthcare needs, and strong base of researchers and professionals. That combination creates growth, challenges, and opportunity for the researchers, medical healthcare sectors, governance. So our question is whether India will participate in the healthcare AI. The more important question is how India can use AI to address its healthcare priorities in a safe, affordable, and inclusive in a practical manner. So for that, we have to think about that how we can use AI in our healthcare sector or medical fields. So it is also highlights that estimated annual growth in AI adaptation across different sectors and the role of India AI mission and investment. But for me, the most important point is not simple market size, but India has an opportunity to develop the solution for Indian healthcare problems. Because India have a lot of population, so we have to take this as a opportunity how to develop solutions for the Indian healthcare problems. Different problems are going on, so we have to think about that. This slide actually talking about a Bangalore as an example using AI powered thermal imaging for non invasive and radiation free breast cancer screening. When we are doing that means different therapies are going on for the different fields. So this kind of example is important because it connects AI with the real across challenges. That means the future of Indian healthcare AI should not be only by about importing the technology from the different regions. So you have to develop the different technologies as per our needs. We could not. That means always we have to import the technologies from the different regions. Must because our population is more, we have to take care of that, and we have to design and develop different technologies for our India or different regions in our healthcare sector, and with the integration of AI. It should also be about creating solutions that are appropriate for our population, our healthcare system, and our resource conditions, different conditions. Because India weather is different and another area weather is different. So to understand that, we must develop the technologies and integrate the AI with that technologies. So it is must our priority to develop the technologies for Indian healthcare system. So this is a example where that means the growth is concentrated. Uh, the chart compares the several segments, including healthcare payers and overall generative AI in healthcare, Asia specific generative AI and agentic AI in healthcare. Then the here the fast moving segment among the categories shown are. So what does agentic AI mean in simple language? That means it refers to the systems that carry out multiple steps a goal instead of simple answering one question. It is a for example, if we will talk about example in healthcare, future systems may support connected administrative and coordination workflows. But there are important principles and the more independent and AI ecosystems, and the more important governance, permissions, and monitoring, and the human oversight. So these are the things we have to take care of, and we must responsible to grow together. So these are that means some microeconomics generative AI in healthcare, how we are growing on, and what are the things that means uses here, and responsible AI ethics governance. When you are using the different AI or generative AI, what are responsible, how to use the AI, what we have to follow the ethics, and how to governance them. So now, in the healthcare sector, the cost of an error can be very high. So according to that, And the technological capacity is not enough. We need trust. That means trust requires responsible design, strong validation, privacy, transparency, accountability, and continuous monitoring of all these things. So these things we will discuss about in the next part. How we'll talk about responsible AI, because in the healthcare, the trust is not capable alone. We have to decide what actually scales into the practice. So you have to use in the responsible manner, how you have to use the AI, and what you have to follow in. You have to follow AI in the ethical manner and positive way. So how we have to consider, that means ethical consideration. That means patients should know when AI is involved in their case. When you are using the AI, the patient must know that we are integrating the AI with those different systems. Should have opportunity to ask the questions. And another thing is accountability. When we will talk about the accountability, if an AI assisted decision goes wrong, who will take the responsibility? If something is going wrong, who will take the responsibility? The institution, or the vendor, or the professionals? So all these things we have to set a framework. Our third question is, if some competence trap is gone, then what will do? If a tool performs well for many months, but people may gradually stop checking it carefully. But we have to take care of all these things, and we have to make a perfect SOP and make a framework, so that everyone can understand all these things, how to use that, and who are the responsible for this, and who are the that means how to use it in a ethical manner. And always the professional must be, that means validator all these things, what are the data inputted, or must be validated. Try to, that means check time to time. Then governance, how you have to govern all these things, how you have to implement the AI for the safe adaptation. Means safely we have to adapt all these things, and it will be easy for all, and that means in the healthcare sector. So AI can assist, the clinicians must decide, but AI can only help. That means assist the what to do, the take the decision, but clinician or professionals must verify it. Is it correct or wrong, and all the validation, every time the validation is going on in the background. Responsible AI is not a one-time approval, but healthcare systems change, or patient population change, data changes, model drift. It must be verified and validated by the professionals. When all these things are going on, it must be validated and verified by the professionals. Therefore, the responsible AI must be. You can say it is a continuous process. Then preparing the next generation, you can say, actually here the slide shows that the gap between the growing importance of AI and the amount of formal AI education received by many medical students. Here we have to move beyond passive lectures. Students must take care of, that means learn through cases, supervised hands-on training, critical discussion, and the real examples. Our goal should not simple to produce people can operate AI tools, but our goal must be to how to produce professionals who can supervise, question, validate, and safely oversees the AI. And you can say, as a whole, responsible use of AI. But the future professionals should be AI literate, evidence aware, and responsible. We must aware that the future professionals must be integrated all these things, and must be literate about the AI, how to use the AI in their day-to-day life, and how to take the responsible, how to validate it, all these things, and he or she must be responsible for that. Then these things are going on now. Over the next 1 to 2 years, the anticipatory greater use of agentic AI and multi-step workflows such as scheduling, prior authorization, and care coordination. These things are going to be done, and over the following 2 to 4 years, that means when we are doing the integration of AI, it will diagnosis by using the AI and radiology into areas such as cardiology, neurology, dermatology. So all these things may be done by the AI when we are integrating all these things. These timelines are not guaranteed, but we may predict all these things going to be happen in next few years. So healthcare and AI are moving closer together. Our responsibility is shape that relationship wisely. We have to use the AI and take the decision wisely, and we have to use the AI in the ethical manner, in the positive way. You have to take in the that means to you can say it is a technology driven. So these are the key about opportunity for the researchers, how to do the things, or that means by using the AI, it may accelerate your work. How to publish the papers using the AI, but you have to use the AI in the ethical manner. And government also released a circular or notice that you have to give the AI disclosure. That means which AI you are using, how you are using, you have to give the disclosure. You have to talk about that. You may acknowledge the that particular AI, which AI you are using. It is a AI disclosure. You have to maintain that. You have to mention that in your paper. I have taken the help of this AI for this. So because it is not most necessary, because everyone using the AI. So AI disclosure is most necessary in day-to-day life also. So these are the key terms we are discussed. How we are that means talking about generative AI, large language model. So these are things we have discussed. These are some references. Thank you very much. If you have any questions or discussion, you may ask. Thank you.
Thank you, sir.
Thank you, ma'am.
Thank you, sir. It was nice presentation.
Thank you,
There
sir.
are a couple of questions. Actually, yesterday also we had, one was presentation by a doctor where they have used the AI model for clinical decision. So one of the concern which I also asked him as well, that how about the data confidentiality? So if somebody is using the generic large language model um, and they are actually giving the data, what about the confidentiality of that data?
Sir, when we will talking about the data, that means what are the how it is private or not, or it is public. When you must take the help of, when we are using the AI, you must ensure that the source of that means the software must be validated and authentic software. First of all, that you have to take the authenticated software. So after that, when you are inputting the data in that software, so that source, that means that source of software, it will ensure you that the data may be confidential, or with you only, not it will be publicly, because AI can use the data for you only, not for the publicly. It is not publicly available. If you give the command, then it will be publicly. But if you give the command, it must be in private and it must secure, I think. But somehow in different stages, it may be violated. But we must take care of that, and we must hire the professionals so that they can take care of all these things.
Thank you. Thank you. It was nice
Thank you.
presentation. Thank you, sir.
Thank you. Thank you very much, sir.
Uh, Thank you very much, sir,
Thank
for
you very
your
much, ma'am.
insightful and inspiring lecture. Your presentation has provided us with an excellent understanding of how generative AI and revolutioning healthcare and shaping the future of medicine. We sincerely appreciate your valuable time and guidance. Thank you, sir.
Thank you. Thank you very much, ma'am.
Yes, sir.
May I leave?
Yes, sir.
Okay,
Thank
okay.
you, sir.
Thank you. Thank you very much, ma'am.
Uh, we shall now begin with our session. Good morning, everyone. Now we shall begin with our oral presentation session. Before inviting our oral presenter, I would like to acknowledge our esteemed panel of evaluators who will be judging today's presentation. Their constructive feedback and scientific expertise will inspire our presenters and ensure fair and comprehensive evaluation process. Our respected evaluators are Dr. Praveen Bade sir, founder of Swalife Biotech, Dr. Pawan Kumar Wankhede sir, assistant professor at Dr. D.Y. Patil College of Pharmacy, Dr. Saranya ma'am, professor at Savita College of Pharmacy, Chennai, Dr. Ramdas Shinde sir, associate professor and head Department of Pharmacy Practice College, Dr. D.Y. Patil College of Pharmacy. Let us extend warm welcome to our respected evaluators. We now begin our oral presentation session. Each presenter will have approximately 12 minutes to present their research, followed by evaluation from our esteemed judges. We respect all presenters to kindly adhere to the allotted time. Now we will start with our first presenter. May I now invite Dr. Mona Lisa Chaudhary to kindly present her work. Hello, Mona Lisa Chaudhary. Am I audible to you?
Yes, ma'am.
Yes, please share your presentation.
Okay. I'm sharing the screen, ma'am.
Yes, please turn on the presentation. Yes, Mona Lisa, your time starts now and ensure
Okay.
you have the 12 minutes to present.
Okay. So, hello everyone. Uh, a very good morning to all of the dignitaries and the judges. I am Dr. Mona Lisa Chaudhary. I'm the assistant professor in Padua University, Goa, and I'm privileged to present a topic on 4th annual Swalife International Conference 2026. My topic is basically one of my PhD works, that is cobalt 2 plus doped biotinylated carbon dot, a theranostic agent for target-specific killing of cancer cells via hypoxia-induced apoptosis. It's already published in Chemistry European Journal at 2023. Uh, Before we are going to our topic, it's a very mostly my topic is about alternative approaches towards killing of cancer cells. It's a very basic study of how can we approach the cancer cells differently um, rather than the conventional systems. So for that, we use nanomaterials as a theranostic agent. That is, it can diagnose as well as treat the cancer cells effectively. I specifically used carbon dot, which is a zero-dimensional nanomaterial, have a size less than 10 nanometer, and it has a very particular property that it is intrinsically fluorescent in the visible region upon UV radiation. Along with that, it have also a characteristics of surface functionalization, which we can do easily. Now, because of intrinsically fluorescent property of carbon dot, and we can module it easily, it is applied in various fields such as sensors, catalysts, and optronics. In biomedicine, it do wonders now. It can be used as a drug delivery agent where simply you add a drug into the carbon dot and it will act as a vector. Or you can integrate something into the carbon dot and became the carbon dot became a drug itself. That's why it will as used can be used as theranostic agent. Now, our topic is approaching the cancer cell treatments alternatively. For that, we all know that there are different kind of hurdles regarding systematic approaches of cancer cell treatments. Chemotherapy, surgery, radiotherapy, immunotherapy, they are a good supporting agents for cancer treatment, but the side effects concerned more and more the researchers and the medical fraternities. That's why new alternative methods with a focus on balance between advantages and drawback of treatment are right now attend the scientists attend the concentration of researchers. So keep that in our mind. We want to approach a different kind of method to kill the cancer cells. We think ki, uh, will we be able to module any biochemical feature that is already present in cancer cells, so that we just module the feature and the cancer cells will automatically goes to the apoptosis. For that, we use hypoxia. It is an important feature of malignant cells that differ from normal cells. Normal cells didn't have hypoxia, but in case of hypoxic cancer cells, the the oxygen concentration in tissues get reduced during abnormal rate of cell division in cancer progression. So it's a biochemical feature of cancer cells rather than normal cells. So it is a differentiation between cancer cell and normal cells. So we thought that what if we module the hypoxic stress in cancer cells and see what happens. So normally cancer cells using the hypoxia as their survival methods. But if we changes its intensity and the duration, the hypoxic stress, then what will be the consequences? For that, we go through different kind of literature and we find that cobalt chloride is an excellent hypoxic mimicking agent. Okay, it will mimic hypoxia in cells. There are multiple research on it, but it is a very toxic substance. So how about we incorporate cobalt 2 plus in carbon dots in such a way that it became more cytocompatible as well as mimic the hypoxia in cancer cells. So cobalt chloride actually inducing apoptosis through PHDS HIF1 alpha pathway, where it simply block PHDS enzyme, that is prolyl hydroxyl domain enzyme, which in present when it's inhibited, HIF-1 alpha protein gets accumulated in our cells. HIF-1 alpha is hypoxia induced factors, and that will generate different kind of pathways, and if we induce it properly and intensify its accumulation, maybe we reach to the apoptosis of cells. So we incorporated cobalt in the carbon dot as a doping agent. We use ethylene diamine and citric acid for carbon core and doped cobalt on it. Then we module its surface with biotin, which is a excellent differentiator between cancer cells and normal cells, because cancer cells have overexpressed biotin receptors. So then we characterized our carbon dot, which have a size between 3 to 5 nanometer in TM and AFM. Then we confirm the presence of cobalt in XPS spectra, and also we deconvoluted our XPS spectra so that we can confirm that cobalt 2 plus is present in our system, not cobalt plus or cobalt 3 plus, because cobalt 2 plus is the only responsible metal here. Next, we go for the amorphous nature of the our carbon dot, which is proved by the XRD, and we have a fluorescence around 460 nanometer. Next, we synthesize another carbon dot, which is just devoid of cobalt, so that if anything, we can compare both of them and we can say confirmly that yes, for the cobalt, the apoptosis occurs, not for the another components. The components are simply there because of the differentiation and increasing compatibility of the system. So we generate same carbon dot devoid of cobalt. They have also TAM AFM characterization having 3 to 5 nanometer size. XPS confirms there is no cobalt. The wavelength is around 457 nanometer. Now we're going to the stability index of our system. So we stabilized it into the media as long as we increase the concentration of FBS for a number of days, and we see okay, a great number of days our compounds are not get precipitated. When we perform the fluorescent intensity test, the photo bleaching property, we observe that it is a stable fluorescent component. Next, we go for the bioimaging. In the bioimaging for both cobalt doped carbon dot and normal carbon dot, we saw that they can differentiate between cancer cells and normal cells. As we know, MDA MB231 is a breast cancer cell and NICT3 is a normal cell. So selective detection of cancer cells could happen by biotinylated carbon dot. Now we're going for the MTT for 2D cell culture. We performed our MTTs and every experiments for both 2D and 2D cell culture and 3D tumors. So for MTT, 24 hours, the cobalt doped carbon dot have 3 to 3.1 higher killing of cancer cells, but for carbon dot, the killing is not significant. Now we are going for the IC50, where MDMB231 and HeLa, both cells are going for the IC50, and we see around 2.5 to 2.7 mg per ml is the dose of our component, which will reduce the population of MDMB231 and HeLa cells for 50%. Now we are going for the apoptosis for both the cells. And this apoptosis shows that the compound goes, the cells goes for apoptosis in early apoptotic pathways. After that, we do a nucleus staining for the cobalt doped carbon dot and for the non-cobalt doped carbon dot. In both the cases, after 12 hours, for in the left side, we see for the cobalt non-cobalt doped carbon dot, the shape and the structure of the nucleus is intact, but in the right side we see they are like crescent moon and the chromatins are condensed into that. So that is the sign of early apoptosis. So in presence of cobalt, this early apoptotic signs are observed, which is not present in non-cobalt carbon dot. Now we are proceed with cobalt doped carbon dot in 3D tumors. We create tumors with the cells in 96 hours span, and it will generate it has a radius around 2 millimeter. Now, when we incubate our cobalt doped carbon dot for 3 days, the 2.7-fold tumor size reduction was observed, and the IC50 for 48 hours and 72 hours are respectively 5 mg and 3 mg per ml. Next, we do a live dead assay for 3D tumor. Simply, the live dead assay observation is concurrent with the MTT assay we saw. That is, the more the day passed, the more apoptosis we see, or the more red fluorescence is present in our tumor cells upon the incubation of cobalt doped carbon dot. And next, we do the apoptosis, where we can see that as the day passed, the apoptosis of cobalt doped carbon dot, apoptosis of cells in presence of cobalt doped carbon dot get increased. Next, we do the Western blotting. So now the question arises in our mind: what is the we know that cobalt follow the pathway of inhibiting PHDS and HIF1 alpha accumulation, but we didn't have any uh, proof of it. So that we do the Western blotting. In Western blotting, we incubated cells.
Mona Lisa,
Yes, ma'am.
you only have 1 minute more to present.
Yeah, ma'am. Just one slide left. We incubated the cells in cobalt devoid carbon dot and with cobalt carbon dot. In the cobalt carbon dot, we saw that the concentration of PHDS band gets faded, and the increase of HIF1 alpha get increased. The band intensity of HIF1 alpha get increased. And in case of hypoxic reagent, we use hypoxic reagent along with nuclear staining, where we observed that as the time passed with the incubation of cobalt doped carbon dot in MDMB 231 cells, we saw more hypoxic reagent color is prominent and the blue color plummeted. So these overall experiments lead us to the conclusion that yes, our cobalt doped carbon dot can modulate hypoxia induced apoptosis and showing high selectivity and efficacy against cancer cells with minimal effect on normal cells. Now, this is a very basic study about our alternative approach of cancer cells, but we can use it for the future application of targeted drug delivery, bioimaging, cancer theranostic, minimal invasive cancer therapy, and personalized medicine. These are my other publications. Thank you.
Thank you, Mona Lisa. Now we will have some round of questions. I respect all evaluators to have questions to Mona Lisa.
Hello. Yes, good morning.
Good morning, sir.
Yes. So, how do you confirm the successful information of this CO2 plus into the carbon dots?
We use XPS spectra, sir. There is a graph, distinct peak of cobalt present
Yes.
in the XPS. As well as we do EDX in same, there also we found cobalt in our carbon dot.
Okay. So, is there any potential toxicity concern associated with cobalt ion?
Yes, sir. Cobalt chloride is itself very toxic. And when we go for the doses, we see that after 5 mg per ml, the toxicity get increased for normal cells as well. That's why we choose a range between 0.5 to 5 mg to do all of our works.
Okay. So any in the current research, is any formulation or anything is available now in market, or it is in the first stage, or any what is the stage of this innovation?
Sir, frankly, for me, it's a chemical biology approach, chemical
Okay,
biology approach. So it's a very basic thing on my side. But as my research are right now, I'm assistant professor. When I do research, I simply try to formulate it into something greater because in vivo studies and comprehensive in silico studies are yet to be remained.
okay, okay. Thank you from my
Thank you.
side. Okay,
Thank you.
over to you, ma'am.
Any other questions from our
Good
evaluator?
morning, Alison. This is Dr.
Hi, ma'am.
Saran here. You were mentioning about use of cobalt in the study against cancer cells, right? And you mentioned that the normal cells were not affected, right?
Yeah, ma'am.
What is your prediction about the pharmacological adverse effects that you would expect if this medication or formulation of yours is brought into the market with regard to hematologic toxicity?
The first thing I have, ma'am, is the dosage because cobalt is a toxic
Am
substance.
I audible?
Yeah, yeah, ma'am. Ma'am, can I answer that?
Hello.
Hello, I'm audible also.
Yes, Mona Lisa, you are audible.
Okay, ma'am.
Am
The
I
first
audible?
thing is yes, ma'am. For the answer, ma'am, the first thing we need to have very specific concern is the amount of cobalt because cobalt is a toxic substance and it is a great concern. So for the drug to formulate, we have to reduce the amount of doping into the drugs so that it will create the essential toxicity around cancer cells but not in the normal cells, as well as we can module the drugs in such a way that they can target specifically approach only towards the cancer cells, not in the normal cells. So if we can balance both of these approaches, I think the toxicity will be minimalized, can be minimalized. Hello. Hello.
Yes, Mona Lisa, you are audible.
Can you hear me? Can you hear me answer, ma'am?
Yes. Yes, yes.
Yes.
I request Ramdas Shinde sir to have some questions.
Good morning, ma'am.
Good morning, sir.
Uh, Ma'am, can you explain any relation of cobalt with polycythemia?
Polycythemia? I am afraid I'm not, sir.
Okay. Can you tell me some serious toxic effect that is
Serious?
concerned with toxic effect?
Uh, for cobalt, the toxicity arises around the uh, ROS generation. If we have profound accumulation of cobalt in our cells, it can generate ROS, reactive oxygen species. So that will also having the capability to destroy the cells for both normal and cancer cells. But yes, the ROS generation of cobalt is very minimal because we do the experiment for that too. So if it is not accumulated in our system in significant ways, then ROS generation is not a problem. But accumulation of cobalt is somehow is a concerning thing for our human body because it's a heavy metal. So it will create toxicity.
Okay, okay.
Thank you.
Thank you, Dr. Mona Lisa. It was nice presentation.
Thank you, sir.
As the work is already published, I would like to give you an opportunity that what would you had change in the methodology if you want to do the research again?
First, sir, I think I would change the system which I use for doping of carbon dot, switching that from carbon dot to biologically gaining molecule. That is, I think there is a molecule right now. So uh, the vesicles or the vacuoles of our cells, which is already present in our cell, and we can use them as a delivery agent. So we can incorporate the cobalt into them so that the biocompatibility will be increased in a significant amount because it is from our biological system. That is the first thing. And the second thing is the amount of cobalt we used. We can also reduce that thing and incorporate another systems so that we can combine the therapies and that will have a greater approach to killing of cancer cells rather than singular one.
Okay, so you are saying that your purpose is to kill the cancer cells, right?
Yeah.
Okay, and what normal cells you have used to compare in MTTSA, in bioimaging, in the 3D tumor
So,
model, Western blot?
for 2D MTT and bioimaging, we have normal cell NICT3 and HEC293.
Why?
The cells, so they are very basic normal cells and they
Yeah.
used extensively for this kind of research purpose.
And why not MCF10A? So our experience working with MDA, that is the breast cancer cells, and MCF7, we normally, when you are using cells, you shouldn't actually have just one cell line of the
No,
cancer. You should have two. Yeah.
we have two cell lines, HeLa cell and MDMB231.
No, I'm talking about breast cancer cells.
For breast cancer cell, we want to use MCF7, but for some reason they can't formulate a good tumor. I think it is problem from our side, as we are not extensively having the expertise of creating tumors at that time. MDMB231 created tumors very easily, but MCF7 they can't form tumors, so we can't do the 3D with them. So we follow the cells. We can do both the 2D and 3D experiments. And MDMB231 is more aggressive than MCF7 and it's the triple negative breast cancer cell.
So which method you use for 3D model?
We used the tumor generation, 3D tumor generation in sir, in the
Hanging drop method.
hanging drop. The hanging drop, sir. We
Okay,
used
so
hanging drop.
one of the question regarding the normal cell line was that MCF10A. Why didn't you use that for the 2D comparison, the normal comparison? Why did you use the kidney cell line and the the other NI1
Red
trait?
cell line. Red cell line. I think that is not available at that moment. I hope
Okay.
MCF10. That's why we don't have the access of that cell line. We have to
Okay, and
buy it.
the last question is that you were targeting apoptosis in the
Yes.
Western blot, or did you do any ELISA or indirect immunofluorescence to focus on a target, a protein from apoptosis?
No, sir. We didn't do that. We just
Okay.
do the enzyme targeting with the Western blotting.
Okay. Thank you. Thank you.
Thank you, sir. Thank you.
Thank you, Mona Lisa.
Thank you, ma'am.
With this, now we will move further to our next presenter. So our next presenter is Akash Podutwar. I request Akash Podutwar to start your presentation.
Hello, ma'am. Am I audible?
Yes, you are audible. Akash.
And the screen is visible, ma'am?
Yes, your screen is also visible. Akash, you will have 10 minutes to present your work.
Okay.
So
Okay.
you can start your work. Your time starts now.
Yes. Thank you so much for giving me this opportunity to present. So myself, Akash Purikar, I am a PhD scholar at Dr. Vishwanath Karat, MIT World Peace University at School of Science and Technology. So topic of my presentation will be nanoemulsion, an therapeutic approach for the treatment of breast cancer. So scientific background, as we all know, breast cancer is one of the most dangerous cancer and most common cancer among the women in India and in the world. So in India, every 4 minutes, one woman is diagnosed with the breast cancer, and every 8 minutes, one woman dies of breast cancer. This significantly tells us the significance of the breast cancer in the India as well as world. So the research problem for the breast cancer is conventional drug delivery systems such as oral and parenteral routes used for the breast cancer. Drug includes tamoxifen, everolimus, doxorubicin, which are associated with the several side effects. This includes nephrotoxicity, cardiotoxicity, hot flushes, embryo-fetal toxicity, hematological side effects, and many more. So the methodology includes uh, binding. So I have performed in silico study in which I have identified the binding affinity of by molecular docking. I have selected AKT1 receptor for that. So it is responsible for the cancer cell regulation, growth, survival, and angiogenesis of the tumor. So in that, I have found that the anti-cancer agent that I have selected was having better binding affinity to the receptor. So the results include pre-formulation study for I have prepared nanoemulsion. For that, I have selected oil component as a Nigella sativa oil, which shows a significant anti-cancer activity in vitro in vivo studies. So it has effectiveness in human. And yet not clinically proven. It has anti-cancer potential in mainly, and its main bioactive compound is thymoquinone. So I have performed GCMS analysis of Nigella sativa oil and found out p-thiamine and thymoquinone are the most prominent agents found in the Nigella sativa oil. I have performed calibration in ethanol, water, and phosphate buffer 7.4. So the pre-formulation of drug includes DSC of the drug, which show two, 212 is the uh, peak, endothermic peak, where the it is the characteristic of the that anti-cancer agent. And so XRD analysis, same analysis showed that it is found in crystalline nature. FT analysis show that the drug, oil, the other component of my system do not have any significant interaction between them. So I move forward to the HPLC analysis. So I have performed ternary plot. In that, I have selected twin 20, 40, 60, and 80. So twin 60 and 80 showed the major plot, sorry, the area. So I have selected twin 60 previously, but in the stability studies, the twin 60 perform uh, gelling. So it was avoided, and we have formulated the nanoemulsion with the twin 80. For nanoemulsion formulation, we have used ultrasonication method, probe sonication method. This is the composition of nanoemulsion. I have performed zeta particle size analysis for that and found out the batch B3 performed better in the particle size analysis, where it reaches to the particle size of 47.09 nanometer. So the turbidity analysis revealed that the batch B1, B2, B3 have the among these three batch, B3 has the highest turbidity as shown in the figure. I have performed thermodynamic stability, in which freeze thaw method is used, ultrasonication is also used. So among the after the thermodynamic stability, the batch B3 performed very well, and it increases the particle size, but which are within the limits, which is 79.70 nanometer. So after that, we have performed drug release analysis through the dialysis membrane and found out that and compare also. I have compared the nanoemulsion with the pure oil. So the pure oil shows the release of 43.55% at the 8 hour, and the prepared nanoemulsion showed 93.74% of drug release at 8 hour. I have also performed DPPH analysis for the which is the radical scavenging assay, and found out oil itself has anti-cancer, antioxidant potential, which is also retained in the nanoemulsion. So after the drug release, I have performed drug release kinetics with the various model like zero order kinetics for first order, Higuchi matrix, Higson Crowell, and Course-Meyer papers, and found out that drug release follows the Course-Meyer papers model significantly. Also, later on, I have performed cell morphological analysis on MCF7 cancerous cell, and showed that after 24 hours of treatment, the cell are pretty much cells are killed. So cytotoxicity analysis was performed by using MTT assay, and it shows the dose-dependent cytotoxicity of the prepared nanoemulsion. Also, cellular uptake as well as the nuclear uptake study was performed by using FITC and DAPI. So it also provided a promising result that the nanoemulsion can get easily incorporated into the cells as well as the nucleus of the cell, where the anti-cancer agent perform its task. So my anti-cancer agent nearly targets topoisomerase 2. So these are the promising results I have found. So after that, drug-loaded nanoemulsion was performed, followed by the release study also performed, particle size zeta analysis also performed for that, and found out it also follows the Course-Meyer-Pappas model. That is, it releases the drug by through the dense matrix of the gel, and found out 96 percent of drug is released at a uh, sorry the I have compared the prepared nanoemulsion as well as gel. So at 8 hour, compared to the nanoemulsion showed drug release of 96%, where compared to the nanoemul gel, it shows the drug release of 87%. These are the references that I have used for the this presentation. Thank you.
Yes, thank you, Akash. I
Yes.
request our evaluators, Dr. Ramdas Shinde sir, Pawan Kumar sir, and Saranya ma'am to have some questions. Am I audible to all?
Yes, ma'am, you are audible.
I request our evaluators to have some questions. I request Praveen Bade sir to have some questions.
Hello. Thank you for the presentation, Akash. It was nice presentation. The same question goes to you, which I asked to the previous presenter. Um,
Okay.
why just use MCF7?
So, sir, the selection of MCF7, there are various cancerous breast cancer cell line,
Yeah.
as ma'am, previous ma'am already tell you that MCF7 upper and MDMB 231. So, MCF7 is non-aggressive and non-invasive. So when we have to study the more metastasis part of the cancer, we generally use aggressive cell line like MDMB 231. But for the non-invasive cancer, we use MCF7.
Okay. And what did you took as a control cell line?
No, nothing, sir.
So how can you say it anti-cancer?
You mean positive control?
Yeah, positive control.
I have took positive control as a paclitaxel, which is standard drug used for the.
No, no. What I'm asking is that the cell study which you have done is on cancer cell line, right? Which
Yes.
normal cell line you have taken to compare the results?
No, sir. No comparative study has been performed yet.
So it will be not used as an anti-cancer, right?
Okay.
Yeah, because if you take paracetamol, right, and if I increase the dose instead of taking 500 milligram, if I take, let's say, 1 kilo of paracetamol, it will be toxic. It will kill my
Yes.
cells, isn't it? Yeah.
Yes.
So it is completely, the cell line study is completely showing result based on the concentration. You need to compare it with the normal cell line. Without that, you cannot say that your drugs are anti-cancer.
Okay. The normal cell line, you mean Hackett or something like that?
Yeah, there is MCF10A, which is again, breast cancer cell line, which can be used as a control. And
Okay.
simultaneously, the other cell line, which Madam have shown in her presentation, all that can be used as a control cell line. You have to use the normal cell line. That is the answer. Because if you
Okay.
take the chemotherapy drug, why they are toxic in the in the treatment? Because the normal cell line is having issues, and that's why the patient is actually having side effect. When you are saying that your drug will be a cancer treatment, you need to compare it with the normal cell line to show that it actually is not only toxic for the cancer cell line, but it is not toxic to the normal cell line. So you have to show that result, isn't it?
Yes, sir. Thank you so
Yeah.
much for that valuable comment. I will definitely look after that. And as this study is not finalized yet and not completed yet, I'm a PhD scholar.
Yeah.
I'm in the continuation of the study, so I will definitely do that.
Yeah, definitely you should include, and also the morphological result which you showed in previous slide. Can you go on the previous slide? Yeah, I don't see anything in this cellular morphology. What actually you have done there?
Uh,
This look white. Everything is white in there. MCF7. So is there anything there? Graph or something?
no, sir, no graph. I was just trying to show that the morphology of the before treatment and after
Ah, okay.
treatment, the morphology has been changed.
Okay. Okay. Because it's completely white, so cells are not seen, properly.
That's
That's
the
fine. Okay.
clarity issues of the image.
Okay. And in the DPPH, what was the concentration you have taken?
Point 1, point 2, point 3, and point 5 volume by percent volume by volume concentration was taken.
How can it be volume by volume? Your drug is in liquid,
Yes, sir, it was oil.
so you have taken what? Zero point 0.2
Point 1,
microliter?
point 2,
Yeah, what? Microliter,
microliter.
ml,
Yes, yes, microliters.
microliter. Yeah, so I think the concentration you are explaining here should be in the, microliter, ml or something, which will be correlating, right?
Okay,
Yeah. Okay. Thank you. Thank you,
thank
Akash.
you so much.
Hello.
I request
Good
Ramdas
morning.
Shinde sir to have some questions.
Good morning, Akash.
Good morning, sir.
Uh, how, how will you demonstrate your formulation induces apoptosis rather than non-specific cell death?
So, for the reformation of apoptosis, we generally perform FITC or DAPI staining. That shows the uh, when the formulation get entered to the cell and it kills the further cells.
Okay.
Generally, after
Overcome.
MTT assay, DAPI or FITC is just to confirm the apoptosis caused by the sample or the drug.
Okay. Okay.
So I now request Saranya ma'am to have some questions.
Yes, please. Akash, apart from the cytotoxicity studies, what were the other studies you have carried out before moving on to the cytotoxicity study?
Uh, just in vitro study has been performed, drug release and all.
Didn't you study anything about the antioxidant activity or anti-inflammatory activity? You directly
Antioxidant
went
study
to the
I performed
cytotoxicity?
at DPPHSA, and
Ah, yes. Okay. Okay. I could see it now. Right.
Yes.
Mhm. Okay. So, what are you planning? How are you planning to extend your research further?
So after this, I've included the drug in this nanoemulsion. So whatever the results have been shown till now are the published are with the oil only. So I have incorporated the anti-cancer agent in that emulsion and formulated a nano emul gel. That will be my final formulation. And after that final formulation, I have performed various anti-cancer studies on the cell line. And after receiving the promising result from the cell line results, I will definitely perform the the in vivo studies, animal study.
Okay. What is the route of administration you are planning for this nano emulsion? If you are
It will be
taking it
topical,
further,
ma'am.
intrathecal. Come again,
Topical,
please.
topical.
Topical.
Yes. So that was the actual hypothesis when I selected my the topic for the PhD. So back then, the tamoxifen was the only drug which is approved, which will be approved, and currently it is in the clinical trial. So it was in the tablet form previously. So scientists have performed the gel of the same drug tamoxifen, and as it was the ease of application for the breast cancer patient rather than taking several injections and several uh, dose of oral tablets. So the drug of tamoxifen was in the clinical trial for, and the same hypothesis I was applied for the selected anti-cancer agent.
Okay. So you're planning to give the drug for the patient after mastectomy.
No, ma'am. When the tumor, so generally it is very selective, and no other study has been performed of similar kind. So I am what I can say. I'm targeting the superficial tumors, not for all types of breast cancer. It will be used as a treatment, but for the superficial cancer, if the cancer is way beneath the what we can say the tissue, it we cannot target it. So the superficial tumors generally can be what we can say show the promising result for the superficial tumor, as it is a gel formulation and it has to bypass several lipid layer of the skin.
Okay, Akash. Thank you.
Yes.
Okay. Thank you.
Uh,
Yes.
thank you, Akash.
Yes, ma'am. Thank you so much.
Yeah. Now we will move further to our next presenter. So our next presenter is Purva Chandorkar. I request Purva Chandorkar to start your presentation. Am I audible to you, Purva?
Am I audible, ma'am?
Yes, you are audible.
Okay. Uh,
Purva, you will have 10 minutes to present your work.
okay.
Yes, your time starts now.
Okay. Uh, Good morning, all. Thank you for the opportunity to present in the conference. My topic is formulation and evaluation of nanoemulgel for psoriasis. I am a PhD scholar at MIT World Peace University, Pune. These are the table of contents. Scientific background. Psoriasis is a chronic T cell mediated autoimmune inflammatory skin disease, and it is a lifelong illness. According to the study, worldwide, the total population has 2 to 3% of psoriasis, and about 30% of people with psoriasis also develop psoriatic arthritis and other comorbidities like heart attack, stroke, cardiovascular disease, metabolic syndrome, depression, etc. Research problem. The conventional drug delivery systems currently used for psoriasis are the parenteral and the oral systems, with some antipsoriatic drugs like acitretin, methotrexate, cyclosporin, etc. which has major side effects like teratogenicity, myocardial infarction, hepatotoxicity, visual problems, etc. Topical formulations have potential to overcome these side effects as they do not reach the systemic circulation directly. And nanoemul gel is one of the such topical formulation which could be used in the treatment of psoriasis. Firstly, binding affinity was studied by in silico study for the active components of lavender oil and my API. The binding affinity was studied with the receptors IL-17 and IL-22, which are the major interleukins involved in psoriasis. Then the pre-formulation studies included FTIR analysis, GCMS of lavender oil, then the pseudo-ternary phase diagram and solubility of API in various oils, surfactants and co-surfactants. Then the same analysis, DSC and XRD, showed that the API is in crystalline form with its endothermic peak at 215 degrees Celsius. Then the nanoemulsion was formulated using lavender oil alone, and the drug release with pure oil as well as nanoemulsion was checked with drug release of nanoemulsion exceeding 100% at 8 hours. It followed Korsmer-Pepper's model, and the DPPH activity showed that the antioxidant potential of the oil was retained in the nanoemulsion formulation. Then the antimicrobial activity performed showed that the lavender oil has bacteriostatic activity. Then the zeta potential particle size and PDI was also checked for the nanoemulsion before and after thermodynamic stability. Among these, batch A3 was selected due to its thermodynamic stability and the zeta and particle size of the batch. Then this batch was further checked for morphological analysis on stimulated Hackett cells. It showed that the treated cells had loss of typical shape of the cells, and it has cellular detachment for the surface, and also has presence of cellular debris which indicated apoptosis. Then the MTT assay was performed to check to get the IC50 and check the cell viability. It showed that batch A3, that is the formulation, had IC50 of 0.4 uh, plus minus 0.04. Then the ELISA was performed for IL-17 and IL-22 interleukins, which showed that the formulation has reduced the levels. Then the nanoemulgel was formulated incorporating the drug-loaded nanoemulsion with zeta potential particle size and PDI analysis. Then the drug content and release was also performed. The release of nanoemul gel was 49% at 8 hours, which showed that it has sustained release. And the drug release, it followed Korsmer-Pepper's model. These are the references. Thank you.
Yes, thank you, Purva. Now we will have some questions. I request evaluators to have 1 to 2 questions to Purva.
Hello.
Hello.
Which biomarker would you evaluate to demonstrate activity?
Sir, further I'm planning to study the biomarkers like psoriasin 1 and beta defensin, which are the prominent biomarkers for psoriasis.
Or any anti-inflammatory biomarker?
Sorry, sir.
With any inflammatory biomarkers?
The IL-17 and IL-22 itself are the inflammatory biomarkers.
Okay. For that, you do the hepatological parameter.
I did not get your question, sir.
For that, you do any histopathological.
Yes, sir. I'm planning to do histopath after the animal study.
Yes, thank you, sir. Um, Now we will move further to our next presenter. Thank you, Purva.
Thank you, ma'am.
So our next presenter is Nikhil Bomble. I request Nikhil Bomble to start your presentation. Am I audible to you, Nikhil?
Hello. Yes, yes, you are audible.
Okay, Nikhil. You have 10 minutes to present your work. Your time starts now. Please go ahead.
Very good noon to everybody now. I am presenting my topic awareness and perceptions of pit and fishes sealants. It's a qualitative and need-based assessment. Myself, Dr. Nikhil Bumble, Professor and Head, Department of Public Health Dentistry, YSMM and RDF Dental College, Ayodhya. So, coming to the background of this study, so the dental caries has been most prevalent and oral disease in children. So 90% of the permanent tooth decay usually begins from patent fishes stage. So sealants introduced to fill those patent fissures were in 1960 and which were proven to be effective, but it has been not so much utilized, so it is in the underutilized stage. So here we try to know what is the awareness and acceptance among the patients, that is the school childrens and the various stakeholders. So that has been remain limited. Moving towards the objectives, so here we are trying to gauge the objectives like the awareness amongst the children, what is the perceptions from the parental aspect, even what are the barriers associated with the school teachers, and even the interns who are going to apply that pit and fish sealants in the teachers. So what are the communication challenges they are facing, and overall from all the stakeholders and even the patients, it will provide us a need based assessment for school oral health, various school oral health programs. So this is an overall gist of this study. So here we initiated first, the research aim here was that we wanted to explore the awareness and perceptions. Then a pilot study was conducted among the 30 participants, and further it was expanded up to the 60 participants, where we achieved the thematic saturation, as this was a qualitative kind of study. So the data collection was carried out with the help of interviews, and focus group discussions were conducted among the school children, among the interns, among the parents, and the other stakeholders. So the data analysis was open and excel and selective coding. So here the research theme which we came up with was the communication gap, and that communication gap was addressed among the children's, teachers, parents, and interns. So this overall give us a gist of a need-based assessment and uptake of the pit and fisher sealants among the school children, and that will help in the reduction of the dental caries. So the results what we achieved over here was that the children were confused with the sealants and the fillings. So fillings are generally the dental fillings, various kinds of dental material is been filled when the patient or when the patient or the children is having a caries. But this pit and fissure sealants are usually applied where the caries has not started yet. So it is the pit and fissures which are present on the occlusal surface of the teeth that are being filled with the sealants, but the children were confused about it and they had an anxiety about the instruments and the pain. The parents were having a concern about the safety issues since the kids were there going to the school, and this was initiated as a school dental oral health program. The school teachers, basically they had a lack of training and knowledge, and they were busy with their daily routine teaching schedule, so it was difficult for them to have that time management and even to put the efforts for the oral health treatment in the school children. The interns who were going and doing the activities under the supervision of the professors and lecturers. So they find simplifying language to explain, like the explanation for the kids in the simplified language was difficult. They were finding difficulties to manage the child behavior. So those were the difficulties which were faced by the interns. And the overall, the emergent theme from this qualitative study, what we gauged, was the communication gap. Coming back to the discussion, so the discussion here is like the children misunderstood the sealant, showing the need for age appropriate education. Then parent express safety concern and trust issues with the school programs. Teachers face time and training barriers to oral health integration. Interns struggled with simplifying technical language and managing the child behavior. Overall, the central theme has been told previously also, that the communication gap across various stakeholders was addressed, and to address these gaps can improve awareness and uptake of the patent fisher sealants among the school children. So coming to the conclusion, the children need to be explained in a simplified language, and they have to be given some visual communication, like how the placement will be done of the sealant material without any pain or anxiety, and in a very smooth and easygoing manner. The parents need reassurance and trust building. The teachers required training and various resources or the literature and how it is going to help the school children. And the interns, definitely they need a support in child friendly communication, as they also have the anxiety while treating the children. So overall conclusion, we would say that it provides a need-based assessment of stakeholders' perspective. It highlights the communication gap as the unifying barrier. It calls for improved communication strategies, teachers training, and parental engagement. So it strengthens the rigor through pilot validation and thematic saturation. So these were my references. Thank you.
What are the
Thank you,
most
Nick.
common barrier you are find in childrens and parents, and for that which measures need to take?
Sir, the common barrier for both of them were the anxiety, sir. The anxiety were most common, and mostly the children were not aware about the safety, but the parents were really conscious about the safetiness of the children, that that material while doing that activity in the school premises would have been really difficult, they felt. But all the measures were taken for that. They were explained the procedure properly. They were shown videos that how the treatment will be carried out, and they will be, it was a minimal invasive technique, like there was no invasion, only whatever the fissures are there present on the occlusal surface will be filled with a material that we called it as sealant material, and it will be given to the children with all the care. So that was explained to them. So they were having a lack of knowledge and anxiety. I found a common issue between them.
Okay, sir.
Hello. Yes, yes, yes.
Thank you. Just one question from my side,
Yes, sir.
Dr. Nikhil. So this
Yes, sir.
program was by a charity or it was by a government program?
So it is an already running school oral health program is given by Maharashtra state government only. But this was like from an institution based we conducted. I am also a PhD scholar, sir. We are doing assessment, and in that study, this is like a study created from that study, sir. Like we were having those stakeholders with us, so we thought that let's uh, create the qualitative part of it also. So we had those people with us, so it was an idea suggested by my guide, so we initiated with it, and
So
we
do you
had.
think that increasing the awareness between the student and the family will actually help for this type of program?
Yes, sir. Basically, what happens, sir, unless and until there is a pain or there is a discomfort
Yeah.
in consuming the food, the people don't take oral health so seriously. And what happens, sir? There is a again misconception that primary, like two times you have a tooth. Uh, uh,
Wash
Eruption, so
clean.
primary tooth is going to go and permanent tooth is going to come, so why to say primary tooth? But if the eruption takes place, sir, at the earlier level, then it creates malocclusion and other deformities further. And if it gets erupted, avulsion of the tooth takes place at that particular time only, then it's okay. But unnecessary, if it gets previously, if it gets avulsed, then students face difficulty. And also, sir, one aspect is that the oral health condition impacts even the learning aspect in the school. So all these things are there. They have they miss the schools, they don't go to schools, they are not able to study. So these are also some of the like qualitative aspects regarding the oral health what we have been learning through literature. So we thought that yes, if that prevention aspect has been highlighted, definitely it is going to help the students as well as it will answer even the burden of the treatment gets increased if this free preventive treatment is not been taken care of at the earlier stage, because economically the parents also land up in burden, sir.
Thank you, Dr. Nikhil.
Yes, sir. Yes.
Hello. Yes, good morning. Can you
If they
summarize your presentation just in a 2 minute, just in a 1 minute?
Yes, sir. This is a study. This is a qualitative study what we have carried out, sir. And the various stakeholders are the part of it. Here we are assessing the Pitten-Fisher sealant application in the school children, and whether they are having any problems, you can say that difficulties regarding it in understanding the Pitten-Fisher sealant. So in that, the qualitative study, in this qualitative study, the most emergent part was the communication gap what we understood over here. So since this was the qualitative study, and the various stakeholders were the part of it, like the school teachers, the interns, the parents and the children themselves, who are going to have this sort of treatment. And we noticed that the anxiety and the safety of the students concerned with the students and the parents was a uh, barrier in delivering this Pitten-Fisher sealant. And the interns were going to place that they were also having a problem in explaining the procedure in a simplified stage. So these were the barriers what we addressed, and we are working on it, sir, now.
Okay. So what will be the future prospect?
Sir, future prospect future prospect is how we convince them and run away the fear, even from the students also and from the parents also. And even we need to bring the sensitization among the school children. Already the government has started working the school oral health programs and started taking the initiation, but still in the rural areas they are not been that much sensitized or aware, or they are not ready to accept it at a huge level.
Okay. Okay.
So only
Thank you
the
so much.
propaganda is a part of it, and making them awareness, spreading the awareness, showing them the videos, sensitizing them through various literature, and even sensitizing the school teachers would definitely help them to allow whoever is in need to get that treatment done.
Yes, thank you. Yes. Over to you, madam.
Yes, thank you, Nikhil.
Yes, I wanted to thank all the presenters, all the people at the backstage also who has taken so much of efforts to conduct this conference. And sorry for inconvenience from my end. Thank you.
Yes. Thank you. So
Thank
now
you.
I request our next presenter. So our next presenter is Prabhat Desai. I request Prabhat Desai to share your presentation. Am I audible to you, Prabhat Desai?
Yes, ma'am.
Yeah. Please share your screen.
Yes, ma'am. Is the slide visible?
No,
Hello?
no, we are not visible to your screen. Can you share your screen?
Yes, I have. Ma'am, now?
No, it's blank.
I'm having a slight internet
Yes,
issue,
now
but
it's
still,
visible.
yeah, yeah. Okay, okay.
Yes,
It might be a little slow. Okay. So
yes,
very good
Prabhat
morning.
Desai.
Yes,
Yes, you
yes,
have 10 minutes to start your presentation.
yes.
Present, so
So
you can start now.
yes, ma'am. So, a very good morning to all the respected judges, and thank you for giving me an opportunity. So, myself, Dr. Prabhat Desai, I am the Associate Professor in Parul College of Pharmacy, Parul University, Goa. And the work which I am going to present now is the research work which has been done by us on green synthesized nano zero valent iron for efficient degradation of paraquat herbicide. So, to introduce paracon dichloride is normally a widely used herbicide, but highly toxic to humans, animals, and aquatic life, and also it persists in a long time in the soil and water, which causes long-term contamination. So, nano zero valent iron is a highly reactive material that, through the literature survey, we have come to know that degrades toxins through redox reactions, and therefore we have thought of coming up with a herbal based green synthesis of a nano relevant iron, which is a sustainable and eco-friendly alternative to the other chemical methods. So the main objective was to assess the toxicity of paraquat dichloride and to check whether the nano zero valent iron was able to degrade it, and also focus on some of the impact on the water bodies using a green herb derived nano zero valent uh, iron. So the main objective was to synthesize NZVI, followed we have planned to use three different plants, that is mint, neem, and lemongrass. So followed with that, we have tried to assess the water quality after adding the paraquat dichloride, assess the impact on Orychromis, and that is the Nile tilapia, that is the fish, and also some of the applications on the environmental remediation. So some of the here, through the hypothesis we have finalized the scope that will help us to examine the toxicity of the paraquat and explore the green solution by using the NZVIs through the phytoconstituents, and also to evaluate them for the contaminated soil and water. So the plants we have chosen, as I've said, is the neem. That's the second, first one. Then second one is lemongrass, and the third is the mint. So we have used a combination of all the three, wherein we have seen through the literature survey. So some of the literature survey was done on individual. So we have thought of using a combination. Maximum of the ones were from the what you can say is chemical based. So we tried some of the literature survey wherein we had found out that it can be used for the degradation and used for preparing the nano zero valent iron particles. So that is how some of the literature survey was done for that. So coming to the methodology, we have used the leaves of all the three plants, and then we have tried to synthesize by adding some of the chemical, that is ferrous sulfate, which helped us to extract the mint, trying to prepare the iron content, mint, neem, and lemongrass. and then addition of sodium borohydrate, which helped at formation of black NZVI's precipitate, which was centrifuged at a high RPM, and then NZVI which were prepared. The pellets were dried at room temperature and stored in a airtight condition to prevent the oxidation. So we assess the effect of green synthesis on Vigna radiata growth. So the soil was collected from a surrounding site from Goa, and try to remove the large particles. Used 15 earthen dishes and washed all the amount of soil, where only the 100 grams of soil was weighed, and 5 Vigna radiata seeds were added to soil and monitored for 7 days. And the soil which was taken was again dried and tested for different parameters. So the different parameters were done for again water analysis also. So we have checked for chemical oxidation demand, we have checked for nitrate estimation, we have checked for nitrite, we have checked for ammonia. This analysis was done also for water analysis. So again done for water analysis was dissolved oxygen and chlorine estimation ion. For soil, we have checked for the moisture content, we have checked for electric conductivity, we have checked for phosphate content, and we have checked for the nitrate content and ammonium content too. So this was the site from where the water and soil was collected from the surrounding site of Goa. So, coming to the effect of paraquat dichloride on physical and behavioral response of Nile tilapia. So, we have checked on the fish, wherein the fish was brought from the area of Majorda, and the fishes were kept in a square tank with 3 fishes per tank, and the concentrations and the control for all. So, 3 tanks were the 0.56 and 12 ppm concentration, and the fourth one was the control tank. So the physical and behavioral studies for 72 hours on the fish was checked. So we have also characterized our NZVIs just to check whether there is confirmation of the iron oxide on the NZVI surface which we have prepared. So the characterization was done where for neem. Once we have done with the neem characterization was done, which confirmed that the presence of functional groups as per the standard. Again, FTIR was done for lemongrass, wherein we have checked again for whether the characterization was correct, and also done for meat. So, coming to the physical parameters, that is for the first one was Vigna radiata, that was the growth of the plant, wherein we have checked the average shoot length, root length, and the health percentage of the plant after the NZVI treatment. So we have come to know the first graph shows you the health percentage, wherein the NZVI treatment significantly improved the health of the plant, with lemongrass achieving the highest health percentage, followed by mint, while the control group showed the lowest at 50%. Coming to the root length of Vigna, again, lemongrass had the longest roots, while the control group showed the shortest. Coming to the shoot length of the plant, the as per the graph showed that the longest was again observed in the shoots growth in the with the lemongrass and ZVI group. So the control plants had the shortest one. So this was the different days and the different growth parameters, and in the pictorial form we also have checked the length and the shoot breadth of the plant. So coming to the soil remediation, so these were the some of the parameters which were first treated before the treatment, wherein we have checked the moisture content, pH, conductivity, ammonia, nitrate, and phosphate, and all these parameters of control plane mint, ZZVI lemon as well, lemongrass as well as your mint and the neem. All the three were again compared with the after treatment. So in a table of form we have checked. So if you see, using the lemongrass have it's showing the best for all the comparative studies. So again, coming to the soil remediation results with NZVI treatment for moisture content. So soil moisture content across different NZVI treatment showed slight increase in moisture content compared to the others, with the highest rise in lemongrass again. Coming to the soil pH, a little shift was observed towards neutral pH levels occurring in all the treatment groups, with NZVI showing the most prominent change from 5.9 to 6.5. So the results for salt remediation for electrical conductivity also was done after the treatment, wherein for lemongrass the electrical conductivity decreased in all the groups, with lemongrass reducing from 675 to 540 microseconds per centimeters. So the graph showed also for ammonia content, wherein the soil before and after were compared, and the results of the treatment showed that the lemongrass showed a clear reduction, lowering from 10.5 to 7.2 mg per kg. Coming to the nitrate content before and after, the again we check the treatments on all the plants. So, and with lemongrass, NZVI showed a rise from 3.3 to 5 milligrams per kg, while mint NZVI showed the highest increase. The phosphate content graph showed increase across all the treatments, specifically lemongrass and ZVIs, elevating levels of phosphate in soils from 10.2 to 12.8. Again, degradation of contaminated water in the control group where no NZVI was added. In the sewage water showed by only 8% of the natural degradation over 60 minutes, indicating limited self-purification capacity. Coming to the degradation of the contaminated via plain NZVI. So the plain NZVI enabled a 32% reduction in contaminants within 60 minutes, while this showed some catalytic effect in the absence of plant-based modifiers, which could limit the nanoparticle reactivity and stability in complex sewage matrices. So coming to the degradation of contaminated via mint, so wherein we checked for contaminated water, so mint achieved around 63% of degradation, showing the excellent, demonstrating the efficiency. Coming to the lemongrass, it showed the highest degradation, that is 65%, indicating a strong potential in removing the pollutants. Degradation with lemongrass achieved around 62% of the contaminant reduction due to its antioxidant properties. So finally, coming to the fish, that is the Nile tilapia, we checked on different concentrations 0 to 0.5, 6 and 12 ppm, and the effect was seen on skin and scales. It was seen on gills. It was seen on the coloration of the body. It was seen on the eyes. Uh, Coming to the fins, there was no damage which was seen on the at 0 ppm, but from 0.5 to 12 ppm, a mild tearing till a major fin destruction occurred up till 12 ppm. For mucus, again layer, there was heavy mucus secretion at 0 0.5 and opaque to thick mucus covering the body at around 12 ppm. So at different hours, also it was checked for if paraquat dichloride on behavior responses. So at 0 hours, it showed a normal behavior. In 24 hours, again for three different ppms, it showed from slightly to disorientation, and at 48 hours, from 0.5 to 12 ppm, from slower swimming, it went from frequent surfacing. At 72 hours, again, they were lethargic, and by 12 ppm, almost they were immobile and floating. Some signs of motility also were checked. So coming to the water characterization with and without paraquat chloride, the COD was very low in without the paraquat dichloride, indicating the low organic matter. This resulted in good water quality, but paraquat dichloride was little introduced therein to increase the COD level, which indicated high organic matter and result in poor water quality. So water characterization with and without paraquat dichloride for nitrate level, where in nitrate level without any paraquat dichloride treatment after addition of paraquat, the nitrate increasing caused stress to microbial activity and also caused oxidative stress to the aquatic organisms. So coming to the nitrate level increased after the addition of paraquat dichloride, indicating stress to microbes and thus disrupting the nitrogen value. Coming to the chlorine levels also after addition of paraquat dichloride was increasing, leading to chlorinated buildup, excess ammonia in water, thus making the water quality poor. And coming to ammonia, it increased after the paraquat treatment, creating oxidative stress and leading to death of the microbial cells. So dissolved oxygen levels over the time Compared to the untreated level, it was very high after we added the paraquat dichloride, and once it was introduced, it depleted even the oxygen levels in water. So coming to the neem, it showed the highest degradation was observed at 12 ppm, followed by 6, and later on was 0.5 ppm. Coming to lemongrass, its degradation showed for paraquat by 53.9% at 12 ppm, and it eventually decreased from 6 ppm to 0.5 ppm. So with for degradation for mint, again the paraquat dichloride, it degraded 44.1 at 12, and it went to up till 27 at 0.5 pm after 60 minutes. Coming to the plain NZVIs, the degradation changed from 37.5 from 12 ppm to 0.5 ppm up till 60 minutes. So finally, I would like to conclude that the demonstrated green synthesized NZVI, particularly from lemongrass, mint, and neem, effectively helped us to in degrading the paraquat dichloride in soil and water. And compared to all the three, lemongrass and neem, NZVI showed the highest degradation efficiency, which also helped us in improving the soil properties and water quality. Helped in sustainable and efficient solution for paraquat dichloride remediation. So in future, we are planning to have a hybrid herb extract which combines with multiple more plant extracts for the enhanced nano zero valent ion synthesis and reactivity, and also studies the molecular effects of paraquat trichloride and nano zero valent ion on aquatic life. So also the impact of this on more aquatic animals also can be studied in future in more detail. So these were some of the references which I have referred. Yes, thank you.
Yes, thank you, Prabhat Desai. I would like to uh, tell Praveen Bade sir to have some questions.
Hello.
Yes, sir.
Yeah, thank you for the presentation. It was nice presentation.
Yeah.
Just one question. Did
Yes, sir.
you actually use the fish or you use the the embryos?
No,
And
we
was it
used that. Yes, sir.
yeah, and was it actually sanctioned by the Animal Ethical Committee?
Yes, initially, sir, what was done was this particular work was done through the animal, because our universities, our college is already affiliated to the university, which itself has an ethical committee. So it was the fishes were brought. The initial proposal was given to the university and ethical committee, which sanctioned the project. Once it was done, at the initial stage, the work had been started, and we have used
Okay,
the fish, sir.
okay, you use the fish. Okay,
Yes.
thank you, thank you.
Yeah.
Hello. Yes,
Yes,
good
sir.
afternoon.
Good afternoon, sir.
Yes. So, madam, how you do select this nano zero valent iron for herbicide?
So, what have we done is through the literature survey, we checked out, we have prepared them actually. So, nano zero
Okay.
iron, we have prepared through by preparing the nanoparticles. So, we have used these three plants, and we have tried to prepare nanos, and then through the characterization, we have checked whether the doping was done or not.
Okay. And why you choose the green synthesis instead of chemical synthesis?
So we already know that we already have a lot of literature survey showing the impact of using all the chemical fertilizers, and what is the impact happening. So in future we will be more studying in details by doing the comparative study. So this work is already in progress, and we are planning to have this natural, because one is thing the thing is that, sir, it is available. These are the ones which are the plants chosen because they are highly available in the state of Goa. So that can be done, and pilot study, if we do it well, scale up, and then commercialization also can be done, because we have a thorough study to prove that the things can be done on a on a large scale mode.
Can you propose the mechanism of this herbicide degradation by NDDVA?
Sorry, sir. How? Why? Sorry.
What is the proposed mechanism of this herbicide degradation?
So herbicide degradation, we initially have done a simple test as of now by checking the thing is that whether the the initial how the, you can say, the chemical degradation can be normally found out. By doing FTIR studies, we checked whether the iniparaquat qualities have been checked. First, the doping has been checked, and later on, we saw that the water analysis, the soil analysis, and on the fish, the degradation happens only on the based on the concentration and the time.
Okay. So is any major limitation to our study?
Not much, sir, but we have. It might be a slow process. Yes, I have to
Okay.
accept it
Yes.
might be a
As
slow
a green
process.
synthesize.
Yes, green synthesis. Yes, definitely. But on a long for a longer note, I can say yes, because we already know that being green, it might be little slow compared, because synthesized one, they have the effect very fast. But we will stick to a project and try to improve the efficiency and try to decrease the time at which it will study. Yes, definitely that note can be taken, sir.
Yes. Yes. Good. Thank you. Very good
Yeah,
presentation.
thank
Thank
you,
you.
sir. Thank you, sir.
Thank you, Prabhat Desai, for your presentation.
Yes, ma'am. Thank you very much for giving the opportunity to the entire team. Thank you so much, everyone. I'll stop sharing, ma'am.
Yes.
Yes, okay.
So now I request our next presenter, Professor Arshia Sultana, to present her work. I kindly request you to share your presentation. Am I audible to you, Professor Arshia
Yes, madam.
Sultana?
Yes, madam. Just give me two
Yes.
seconds.
Please. Yeah, okay.
Yeah. I'm sharing my screen, madam. Just give me 2 seconds.
Yes, yes.
Very good afternoon to all of you, respected dignitaries. I think so. My screen is seen. Excuse me.
Yes, yes, it's visible. Can you please turn on the presentation mode?
Okay.
Yes.
Very good
Yes, it's
afternoon to all
visible.
of you, respected dignitaries, judges, organizing committee members, delegates. Foremost, I like to thank the organizing committee members for giving this opportunity. Thanks. And today my presentation is on machine learning algorithm for classification of treatment outcome in uncomplicated pelvic inflammatory disease. My co-authors are Dr. Sumbul Muhammad Bilal bin Hayat. He is working in China and he has done the machine learning part of this study. So here, disclaimer, this Unani literature has been taken from the classical text and the modern publications has been seen for the recent advances and we have made these images with the help of ChatGPT. With this, uncomplicated pelvic inflammatory disease is an ascending infection which occurs in the upper genital tract through the vagina and enters through from the vagina it enters to the cervix and to the cervix to the whole pelvic cavity. So what we have seen that the laparoscopic studies have said that the incidence of uncomplicated pelvic inflammatory disease is around 30 to 40% of cases, and most important, this is not caused by a single microorganism, but it is a polymicrobial in etiology. It can be caused because of gram negative or gram positive organism as well as fungal infection. So we see that a number of organisms can cause uncomplicated pelvic inflammatory diseases. When we say pelvic inflammatory disease, it can be mild, it can be subclinical, or it can be severe. So we see that mild or subclinical PID has mild lower abdominal pain, occasional discomfort, and sometimes this infection may not have any symptoms or it may be say, asymptomatic. So we see that this PID, pelvic inflammatory disease, is also called as PID, which is a major pelvic health problem and has emerged as a silent killer that disturbs women's life, which leads to various complications because it can be caused by sexually transmitted disease also. The cases are rising in developing countries such as in India, and the exact incidence of PID is very difficult as the gold standard diagnostic is laparoscopy. So here we have seen that as the PID has become a global health problem, and we know that we are already facing antibiotic resistance with many antibiotics, and we are planning to go for AMR, all that thing. But we see that compliance with antibiotic therapy for PID is very poor, particularly in adolescents and in those receiving complex prolonged treatment regimens. Further important thing what I like to say is it is having higher risk because it is having high increase in the resistance levels of pathogenic bacteria, multi-drug resistance in bacterial pathogens. And there is sharp decrease in the efficacy of conventional antibiotic therapies as there is increased load of disease in the population. So when because what is the rationale of the study is that as we are seeing that most of the bugs are becoming resistant to the antibiotics, so now we are seeing that many of the researchers as well as common public is turning towards complementary and alternative medicines, and Unani medicine is one of the complementary and alternative medicine which works on herbs as well as minerals and animal drugs. So we see that in classical Unani texts, there are many herbal drugs which can be given in PID like Plantigo, Solanum, Chinchorium, Trigolina, Beriberi, and others. So here what we have done, we have selected a pharmacopoeial preparation from a Unani text where we can use sesame seeds, black sesame seeds and rose oil. Together, we can give cotton suppositories of pessaries, which are useful in uterine inflammation. According to Unani concept, these drugs are having anti-inflammatory, analgesic, and other beneficial effect. Both the drugs. And then when we search the literature, contemporary literature, we were able to see that this black sesame seeds are having antimicrobial effect and are able to act on multiple microorganisms. Not only they're antimicrobial, but they're also having anti-inflammatory, analgesic, and antioxidant activity. Likewise, rose oil is also having anti-inflammatory, analgesic, and astringent property, and it is again having effect on many bacteria together. With this antimicrobial, anti-inflammatory, analgesic, and antioxidant property, these drugs were selected and the medicine was given per vaginum to the patients. Then, along with this, what we did, whatever the results we got or whatever the data we have received, we have went for machine learning for clinical decision making. So we see that now we are seeing that machine learning is an emerging and promising tool for some more supporting clinical decision making. So what we did in this study, we have applied the AI technique, predominantly ML models, to classify experimental and standard groups. So the classifier used were AB, NB, and DT in this study, and then we have trained it using the post-treatment and classified into treatment and control group, and leave one out cross validation was used to evaluate the model performance. So this was the machine learning what we have applied in this study. So with this, the objective was to determine the therapeutic potential of sesame seeds with rose oil in UPID, that is uncomplicated PID, and second objective was the to perform different ML algorithm in classifying treatment groups of women with uncomplicated pelvic inflammatory disease using clinical outcome. So basically, this study was a clinical study, and then we have applied the machine learning. So the study design was comparative, prospective, single-center, double-blind, double-dummy, single-blind randomized standard control trial, which was carried between February 21 to January 2022. This trial was registered in clinical trial registry, and the sample size was 60. According to the previous study, we have calculated the sample size. It was a single center done in National Institute of Unani Medicine. Before starting the trial, ethics approval has been taken. The trial was registered in CTIR, ICMR CTIR, and then after that we have started the trial, and you have followed the GCP guideline to do this study. Then the inclusion criteria for the study were married women with 18 to 45 years of age, diagnosed cases of pelvic inflammatory disease with complaint of adnexal or cervical motion tenderness, lower abdominal pain, vaginal discharge, low back ache, and features of UPID, that is mild or subclinical PID. Then the exclusion criteria was complicated cases of PID or any conditions likely to require surgical intervention within 24 hours of the start of the treatment, women with any malignancy or systematic diseases, women on OCP and IUCD, pregnancy and lactation, delivery and abortion within last 3 months. These were the exclusion criteria of our study. Then the interventions were, as I said, it was a randomized standard control study. The intervention group included, as it was a double dummy study, so orally we have given placebo in here, and locally we have given vaginal pessaries of rose oil with black sesame powder, whereas in control group, orally we have given antibiotics, that is doxycycline 100 milligram twice daily and metronidazole 400 milligram thrice daily for 14 days, and locally we have given placebo for 14 days. So here, according to the method of preparation of the drug, the drug was prepared on the basis of standard Unani pharmacopoeia, and the route of administration, already I said, orally placebo is given and vaginal pessaries was given for 14 days. So this treatment protocol therapy was for 14 days. Then the primary outcome was clinical features that included VAS score for abdominal pain and low back ache and McCormack pain scale for tenderness. The secondary outcome was puzzles and wet mount test and health-related quality of life assessed with SF12. As I said you, the sample size was calculated from the previous study with statistical power 20%, level of significance 5%, and 10% dropout. The total sample size was 60. Sampling technique was simple random sampling. Randomization and blinding, allocation was 1:1, which was generated by the computer generated randomness. A single block using a single sequence of random assignments were taken. Concealment, the sequence was concealed from the researcher until the interventions were assigned using an open list of random number. Blinding, participants were blinded here. Masking and matching, the test drug and standard was filled in the identical color capsules, and locally we have given palm sugar as a placebo. Statistical analysis included two-sided p-value with type 1 error as 0.05 with 95% of confidence interval. We have done intent to treat principle following last observation carried forward method, and according to the data we have used for normally distributed data, we have used paid and unpaid student t-test, and for squeeze data we have used Wilcoxon's match pay test and Mann-Whitney U-test. This was a diagnostic tool which was used in these cases, that is visual analysis scale for pain, McCormack pain scale for to assess direct and report tenderness, and Wetmont for puzzles, and SF12 for quality of life. The results, as we have seen the flowchart, a consort statement, flowchart of participants, we can see we have seen 110 cases in which only 60 persons were randomized and 96 patients were eligible, and they declined to participate with 6 and excluded with 30. So each group had 30 patients. There was around 3 patient dropout in intervention group as well as 3 patients dropout in the placebo group, standard group. So and then VAS and pain score over the time, we can see that on day 0 the VAS score for lower abdominal pain and for low back it was 2, and slowly we were able to see on day 15 it is decreased to 0.36 to 0.4, and same thing we can see for low backache it was 1.16 to 1.14. So we were able to see that both the groups, because we have taken standard here, both the groups showed decrease in the pain on 14th day. And again McCormack pain tenderness scale also showed that there was decrease from 5.76 to 5.76 in test group and 5.6 in standard group. It decreased to 0.93 to 1 point, and in standard 1.03. So the results was non-significant between the group because we have taken standard. Both groups were effective in controlling the pain and tenderness. Then the secondary outcome was SF12 quality of life. We can see that physical component score was in both groups. It was increased at 14th 30 day, because we have taken after 1 month what was the quality of life, because 15 days was observation to see that there was any recurrence or others. So we were able to see that there was increase in the physical component score as well as mental component score, and the total score also increased at 30. At baseline, it was 58.6 and 58.7 58.47 in the intervention group, and sorry, 58.6 is in the intervention group and 58.47 at baseline. And we were able to see at 30 days the quality of life has improved drastically. It was in intervention group, it was 82.79 and 80.04 in standard group. Both were significant with p-value 0, p-value more than 0.05, showing that both groups were equally effective in improving the quality of life. Then we can see wet mount test WBC count at baseline. The WBC count was 31.21 in the intervention group, and it was 29.7 in the standard control group. At post test at 15th day, we were able to see that there was drastic decrease in the WBCs in vaginal wet mount test to 5.7 in the intervention group and 8.1 into the standard control group. And when we have seen the safety profile of both the groups, that is as your liver functions and as well as renal function, we were able to see that the drugs were not having any adverse effect on the lungs and the on the liver and the kidney, with p-value more than 0.05 in all the safety profiles. And here we can see that it was comparable with the standard. Both drugs, the standard group as well as the intervention group, both were equally effective, showing p-value more than 0.05. But pre and post test, when we compared, there was an extremely significant difference with p-value less than 0.001. Again, then machine learning classifier was added to this, and we were able to see that NB leave one out was high, showing the highest value of 0.7405 AUC precision and accuracy. Recall, we can see that the best classifier was NB in this study. Discussion. This proves that intervention was as effective as standard rig in elevating symptoms of UPID and thereby improving the health-related quality of life. Safety assessment showed no adverse effect were reported in both groups. So here we can see that as we selected this drug based on a Unani ethnomedicinal uses along with the pharmacological activity which was done in preclinical study, we were able to see that these were having anti-inflammatory, analgesic, antioxidant, antimicrobial, hepatoprotective, nephroprotective, and antiproliferative activity of black sesame seed as well as rose oil. So with this, we were able to see that as the rose oil and black sesame seed both are having phenolic compounds which have flavonoids, terpenoids, and others which have antibacterial activity, and the active phytoconstituent has been listed here, which are able to act on polymicrobial organism. We are able to see that they are having effect on many organisms. Therefore, we can say that these drugs may be effective in treating UPID. As I said in my first slide, that usually the pelvic inflammatory disease is caused by polymicrobial organism. Likewise, black sesame also have phenolic compounds which are having lignins, sesamin and sesaminol, which have already been proven preclinically in the as an antimicrobial activity. Not only this, these drugs are not only having antimicrobial effect, but they are also having anti-inflammatory activity. Like for example, we can see that sesame and sesamon, because we see that in PID, because of this microorganism infection, it causes inflammation. It causes inflammatory insult, causing increase in tissue injury, and therefore there is release of pro-inflammatory mediators like IL-6, TNF, IL-1 beta, and NF, KB, and INOs. So when these all pro-inflammatory mediators are released, this active phytoconstituent, that is sesamin and sesaminol, and in rose oil we can see this phytoconstituent which can suppress the pro-inflammatory mediators, thereby inhibit the pain. So we are seeing that indirectly it is able to decrease the pain because it is having anti-inflammatory property, and it acts on the this pro-inflammatory mediators. And not only this, we can see that the pain sensitization is carried by the central sensitization and peripheral sensitization, causing lower abdominal pain. So we are able to see that it is not only inhibiting the pro-inflammatory mediators in the tissue injury, but also it is also able to inhibit the central sensitization of the pain. By this, we can say that the rose oil and sesame seed are helpful in decreasing the inflammation. Then it, as I said you, these are also having antioxidant activity. So they are able to, because of the antioxidant activity, they are able to decrease the oxidative stress and
Arshia
thereby.
Sultana?
Yes, madam. It's
Yes,
over,
your
madam.
time is over. Yeah.
Only two slides are there. So we are able to see that NB model is more appropriate for classification of intervention and standard group. And my strength and limitation is till date none of the clinical intervention studies have evaluated the effect of rose oil and sesame seed in UPID and health related quality of life. Second standard, it was double dummy single blind randomized standard control with intent to treat, analysis and good compliance with only 3 patient dropout in each group. Limitation was we cannot extrapolate this to acute inflammatory diseases considering the severity of disease and limitation of the trial. So conclusion is that our finding indicates that cotton pessary of black sesame with rose oil has a therapeutic similar effect, and again we can see that it enhances the health related quality of life, and most important is that they are phytoconstituent which helps in the decreasing the inflammation. When in machine learning, NB classify easily classify both groups of PID. In addition, we would apply our classification method to other female infectious diseases. Thank you.
Yes, thank you, Arshia Sultana. I request our evaluator, Pavan Kumar Wankhede sir, to have some questions.
Yes, good afternoon. Yes, good afternoon, Sultana ma'am.
Good
So
afternoon,
my
sir.
question is, how exactly did you define treatment success and treatment failure in your database data set?
So we have seen that, uh, like a success rate, we can see we have seen with machine learning. So what we were able to see that 60% patient had got good response, and when we have compared, like we have only compared the this one like a VAS score, and we were able to see that we have calculated the percentage improvement also. So the percentage improvements were very good in both groups. Like we can't say it was 100%, but both the groups were more than 60 to 70% response in both groups.
Okay. So which clinical variable control contributed most of the treatment failure, and how do you determine their importance?
So here we have not seen the confounding variables because main target was to see the efficacy of the drugs. So we have seen the
Okay.
efficacy of drugs, and they can be confounded. Like for example, patients if they have menstrual periods, they may not get the response. So that confounding variables we have not seen.
Okay. So why is machine learning appropriate for predicting the PID treatment outcome?
It was FSC, so we have seen, we have AUC, we have applied the precision, everything. So we were able to that see that these results were 68%, they have given the precision. That means we can say that these results are more appropriate because higher value shows that the efficacy was good. So with that, we were able to say that whatever data we have seen, we have not biased the study, and we were able to say that we got 68 to 70% response. And the same thing was proved by the machine learning that the precision and recall all that have shown that it was around 0.78, that is 78% length. In NB have shown
Okay.
that classify have shown that there was 78%. Whatever the results were there, it was accuracy was around 68 to 78%.
Okay. So as a machine learning, how will you externally validate your machine learning model before using in clinical decision?
So actually, machine learning. I'm a clinician. So I have given
Okay.
my
Okay.
data to the person who have no machine learning. So he's a co-author in this, and I like to thank him very much. Because of him, I was able to understand what is artificial intelligence and AB, everything, decision tree, all that thing. So he has done this machine learning, and now he's not feeling well. So I was not able to discuss many things with him regarding
Okay.
this.
Okay. Okay. No problem. But this is the means if this is a good way, but is it you can validate this for the
Yes, we
clinical
have
outcome?
actually tried to validate the clinical outcome with the machine learning, and what we were able to see that because it was a single blind study. One of the limitation was it was not a double blind study, so it was a small sample size also. So we were able to say that okay, we have done it's a clinical efficacy. We are saying that it is around say 68 to 70%. Same thing was proved with machine learning, say stating that whatever the efficacy outcome was there, the same efficacy can be we have seen with the machine learning.
Is that this model will help the clinician in PID management?
Yes, sir. Yes, sir, it will help because, as I said, we can't say any drug will not have 100% effect, but we are able to say that, and because we have compared this study with the standard, we have compared the standard what is given in the in the conventional medicine, that is doxymetrozole. It has been said that it is a gold standard treatment for PID. This gold standard treatment we have compared with the efficacy with our medicines, and we were able to say that they were the this test intervention group was comparable with the standard group. So we can replace the antibiotic with with our herbal drugs, and with this we can prevent antibiotic side effects, because we see that when we give metronidazole they will have metallic taste, and again the immunity will decrease, whereas when we are giving this local medication, the drug is having local effect, and we are able to prevent the side effect of the drugs, because
Okay.
when we give antibiotic they will be all disturbed in the flora of the gastric flora will be disturbed, and then 14 days patient has to take the antibiotic, and we are able to see that many of the microorganisms are becoming resistant. So with this plan we thought, let us take a local vaginal application and let us see what is the effect of local vaginal application with the antibiotics. So when we compared the study, and we can see that in secondary outcome we have kept the WBC count also. So we were able to see that WBC count was decreased in both the groups equally, because we were able to
Okay.
see that there
Okay.
was a significant.
Okay. 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 It's okay. Can what's your name, Doctor Arshana?
Yeah, yes.
Okay. It's over from my side. You can yes, go to the Ruchida madam.
Oh, yes, thank you, Arshia.
Thank you, ma'am. Thanks for giving this opportunity.
Yes, thank you.
Can I stop sharing, ma'am?
Oh, yes, yes. Now I like to welcome our next presenter. So our next presenter is Deshraj Chumbare. I request Deshraj Chumbare to start your presentation.
Yes, good afternoon.
Yes, yes, please share your presentation. Are you able to upload?
Yes, so that processing is going on.
Okay.
We uploading now.
Yes. Okay.
Yes. Can I
Yes,
present,
now.
madam?
Yes, okay. Okay, Deshraj Chumbare, you have only 10 minutes to present your
Yes.
work. Your time
Yes, yes.
starts now.
Okay. Good afternoon to all. Myself, Dr. Deshraj Sumre, Associate Professor, Department of Pharmacognosy, Amrutwani College of Pharmacy, and I am going to present my presentation on phytopharmacognostic profiling of the abenomious manihot medium. I have go through the traditional, that is the number of medicinal plant herbs, and that traditional herbs. Recently, I have found that this medicinal plant, that is the Abedomyces manihot, it is a very effective for wound healing activity. So generally, I go through the practically, that is identification and their quality standards for their standardization. I have explored this plant for getting the pharmacopoeial standards, also for their exploration for their standardization for quality and purity also. These pharmacopoeial standards have including the number of parameters, including morphological as well as anatomical characteristics, and as well as physico-chemical standards that have included into this. So in this present investigation, this Abelomosis manure plant I found or abandoned in the Trimbakeshwar Hills, and traditionally this plant stem powder, it is a very useful for the number of medicinal elements. So in that, especially that is a wound healing property, this plant it is a very useful one. Botanically, this plant is a also name is a hibiscus manure belonging to the Malvaceae family, and in vernacularly it is called as Jangivindi. And this plant is a, it is a that is in Konkan regions, especially also into the western Ghats of India. This plant it is a form. Okay. Literature, when I go through the literature review, I find that this plant it is a, having the importance into the useful as a immunobalm and also treatment of the wounds and cuts. Also, it is a very useful for the boils, sprains, inflammations, tuberculosis, and leukoderma. Also, it is useful for the chronic bronchitis and headache. This plant it is very importantly showing that it having the presence of the steroids, sigma sterols, cytosterols, as well as heparosides and flavonoids present in these plants. In this plant, pharmacologically, it is an anti-inflammatory property, also bone sparing activity, neuroprotective, antiviral, wound healing, and larvicidal activity. So, in the present studies, I have taken these plants to perform their first authentications, then also morphological, exomorphic characteristics, histological, physicochemical standards of the Abelomosis stems. And these plants also go through the process of the collection, authentication, processing, and laterward phytopharmacognostical properties by evaluating their the stem portions, and it is authenticated from Department of Botany, that is in Sangam. Also, then later what it is a prepare the powder. After preparation of the powder, it is a process for their preparations. So this one is a plant image, and that I have prepared that this herbarium also. After preparation, the authentications have done, and after authentications, I go through the morphological external characteristics observation, that is a color, odor, taste, size, shapes, as well as their sectioning, histological examinations of the stem portions they have performed, and stain with the fluoroethanol plus concentrated hydrochloric for the lignified cells. Also treated with the acetic acid for observation of the calcium oxalate crystals and observed under the microscopes. So go through the also similarly powder characteristics by clearing, staining and mounting and observation methodology. Also, I have done that is a physico-chemical evaluation such as ash value, acid soluble ash, water soluble ash, sulfated ash, alcohol soluble extract to value, water soluble extract to value, moisture content, and total crude fiber content. Also done the fluorescence study. I observed that powder with the different solvents under the UV cabinet, short and long wavelengths. Also, heavy metal and minerals, it is according to the WHO guidelines. This parameter it is also essential. So I go through the heavy metal and mineral analysis by prepared that powder, fine powder, and reflux for reflux with the that is nitric acid. Then also that is added the hydrogen peroxides, and then it is a filtered and cooled, and then it is analyzed under the atomic absorption spectroscopic technique. Also prepared the ash of the same powder and uh, analyze that stem powder. Okay. Then extractions have performed, continuous extractions have done, continuous hot percolation methodologies, oxalate extraction is used for extraction. Firstly, that powder is defatted, then later that powder it is extracted with the methanol, and then methanolic extract again it is a fractionated with ethyl acetate soluble and insoluble portion. You see this graph. So by this way flow chart, but I go to the coarse powder, petroleum ether extract, then petroleum ether mark again is taken and it is extracted with the methanol, and then methanolic extract fractionated with the ethyl acetate soluble and insoluble portions, and also that remaining mark is extracted with the aqueous. Then also done the number of preliminary phytochemical tests as per the WHO guidelines. I observed go through the that is the test alkaloid, glyphosate, flavonoid, tannins, phenolic compounds. I check their presence under these plants. Also morphologically, I have observed that this plant is having yellowish color flower, and that is a Malvaceae family drug. So it is having tripolar type of the shape of the leaves, and stems that is having the crimson red color, and that stems also that is inside that is yellowish, externally that is a crimson red color. Characteristics odor, bitter in taste, and their size is a 3 to 6 centimeter high, and so straight erect type of the that is a stem. Then also I go through that is a microscopical examination, so observe the that is a cork, phylum, phyllogen, and phylloderm, and also secondary phloem portions. This is a diagram, so we can I got this picture also. That is a cork, then secondary phloem portions, that is a phylum, and then also observed that is the vessels, so xylem vessels into the pericyclic portion of the stems. Then go through the powder characteristics. I observed these lignified fibers as well as white fibers, and the star in blue color, star green, and acetic acid. When treated with that, I observed the insoluble type of the calcium. Also go through the physicochemical studies. So I observed that that is this number of physicochemical standards, and it is a very helpful for maintaining the quality standards as well as checking and evaluation of the inorganic salts by the ash values as well as extractive value helpful for showing the chemical constants, and also it is a moisture content to check the moisture presence of the moisture, and food fibers that is the presence of the fiber content. Also, this is a behavior analysis means chemically. So what they observed, and it's showing the presence of steroids, triterpenoids, also tannins, and alkaloids and flavonoids is present in these plants. Then I have go through the heavy metal and mineral analysis also. So it observed that this lead, aluminum, magnesium, zinc, copper, iron, and nickel, chromium is also having abundant in these plants. Aluminum not detected. And these extractions I have done. After extractions, I got this extractive value of petroleum, methanolic extracts as well as ethyl acetate soluble and insoluble portions and aqueous extracts. So this study have also shown that presence of this petroleum ether showing the steroids and triterpenoids. Ethyl acetate fraction, especially soluble fraction, showing the presence of the flavonoid and rich amount of flavonoid, and methanolic extract also showing the phenolic presence of the phenolic compound, and aqueous extract showing the carbohydrate glycosides and also phenolic compound. So, concluding that this pharmacognostic and phytochemical standards of this plant, it is very helpful for compiling the quality control standards, and this extraction indicated that presence of the chemical portions that is a very beneficial for the number of biological and pharmacological properties. So it is having important for the compilation of this data for the identification, proper identification, authentication, avoiding the adulteration, as well as quality control standards. And it is very helpful for compiling the monograph of these plants. And also futurely, it is helpful for preparation of the any one of the formulations. Also, that is a chemical, phytochemical analysis of detailed phytochemical analysis of these plants. So it is these are the references I go to that. Thank you. Thank you for listening.
Yes, thank you, Desha Sumre. I request our evaluators, Dr. Pawan Kumar Wankhede and Dr. Praveen Badit, sir, to have some questions. Am I audible?
Hello. Yes. Uh, can you explain your topic in the brief 2 minutes?
My topic actually phytopharmacognostic compilations of these plants. It is important for identification, authentications, as well as compiling the quality control standards of this abedomyces manual. It is that is a jungly vendi. That is one of the jungly. This plant is called as, and this plant it is a very medicinally important for the wound healing property. And that is a locally it is a useful.
Okay. Can you go to the first slide?
Yes.
Kalpana Devi. Okay, phyto pharmacochemistry profile of that particular drug. Okay. So which pharmacological activity this and which phytoconstituent is responsible for that particular activity?
So this plant is when we are going through the literature. So I found that this plant it is a very beneficially for wound healing activity. And ethnomedicinally, this plant it is a useful for the immunobox as well as also boils and all that treatments. And this plant, it is a chemically, phytochemically consisting of the steroids, especially beta cytosterols, gamma sterols present in these plants. And flavonoid is also present in these plants. Leukoprotein. So this plant is having phytochemically also strong. That is steroids, triterpenoids, as well as flavonoids present in this plant. And pharmacologically also it is useful for the anti-inflammatory activity, wound healing activity, and therapeutically important.
Okay, which part of the plant are most useful?
Actually, stem it is a very useful one because stem powder traditionally here local peoples is also in Maharashtra utilize that powder for the that is a property of the wound healing. So on that basis, I have collected this compiled this information, and this
What will be
plant is
the
a
any
one.
future prospect of this? Which activity you want to do in future?
This is actually on this plan. I have taken for the next step for going to work as a preparation of the any herbal transdermal patch because this warm healing property also an anti-inflammatory property, but no any that is another formulations have prepared any type of the formulations, and that patch herbal patch it is a very helpful for next anti-inflammatory actions of this
Okay,
plant.
if you want to isolate that particular product from this plant, so which method you will select?
Actually, I have go to the preliminary phytochemical work,
Okay,
but
so you can go for the isolation and this you can go to the that particular
this
column
is by
chromatography or flash
column. We
chromatography.
can easily perform by the column separations also, by
Okay,
column extractions, and by this way we can fractionated and isolated that material.
okay. Thank you, Dashraj. Over to you, ma'am.
Thank you.
Thank you, Dr. Deshra sir. Nice
Yes, yes.
presentation.
Thank
Just one
you.
question. So, what type of extraction method you are using?
Actually, I have go through the continuous hot extraction process by using the oxalate extraction. And according to that continuous extraction methodology, successive extraction methodology, firstly, powder sample is a defat. So defat by using the petroleum ether. After defat material, then we can forward for the next extraction while for the methanolic extraction. And this methanolic extract we can also using the fractionation separate out the flavonoid portion by into the ethyl acetate soluble fraction. So that ethyl acetate soluble fraction containing the flavonoid easily solubilized we need to separate out.
So, the major group you are focusing is flavonoid.
Yeah, because already that plant is having that colored compound because this after extraction that is indicated that extract also, and that portions it is a very flavonoid it is having that type of the biological potential, so it is a focus on that similar.
Okay. Thank you, sir. It was nice presentation. Thank you.
Thank you, sir.
Yes, thank you, Desha sir. Thank you, sir. So now I would like to invite our next presenter. So our next presenter is Shalini. I request Shalini to start your work. Please turn on your mic. We are not audible to you, Shalini. Am I audible to all?
Yes, you're audible, but Shalini's voice is not there, so I think her mic is not connected.
Okay. Shalini, please turn on your mic.
So, Shalini, when you click on the link, you have to give the permission for mic and the camera. So, I think you have not given permission for out of that, and that's the reason your device is not connected.
Okay, we will wait for some minute till Shalini joins. Shalini, you are not audible to us. Your mic is off.
Ruchira, I think we will take next.
Okay, sir. So our next presenter is Rupali Nath. So I request Rupali Nath to share your presentation. Rupali Nath, can you hear me?
Good afternoon.
Yes, please share your presentation.
Yes, ma'am, I'm doing that.
Yeah.
Is it visible?
Yes, it's visible. Oh, yes, Rupali Nath, you have only 10 minutes to present your work. Your time starts
Okay.
now.
Okay, ma'am.
Yes.
Thank you. Good afternoon to all the respected dignitaries present over here, respected evaluators, and all the participants. Myself, Rupali Nath. I am a PhD research scholar in computational biology and bioinformatics laboratory, Barampur University, Odisha. So, is it visible?
Yes, it's visible.
So my work is in silico compound as dual inhibitor of LASER and RLHR current sensing receptors in Pseudomonas aeruginosa. So these are the contents. Pseudomonas aeruginosa is a major opportunistic multi-drug resistance gram-negative bacterium and is mentioned by a critical priority pathogen by the World Health Organization because of the biofilm formation property and the virulence mechanism. And the virulence, motility, and biofilm formation, all these things are regulated by a cell-to-cell density-dependent signaling method, that is the quorum sensing. And LASER and RLHR are the central transcriptional regulator of this quorum sensing mechanism. In the right side, we can see the whole mechanism in Pseudomonas aeruginosa. It begins with the last system, which starts with the synthesis of some signal molecule, that is N-acyl homoserine lactones. It's synthesized by the last eye, then it came out of the cell by certain explosive pumps, and after reaching to certain threshold concentration, it again came back to the cell and activate the LASER receptor. And in response to that, the LASER receptor activates many downstream signaling system, for example, RHLR and PQS system. Along with that, it also activates many proteases and elastases that helps in the tissue degradation of host cell and helps the pathogen to enter into the host cell. Similarly, the RLHR system also activated by the RLHI gene by synthesizing the signal molecule, that is butyryl homoserine lactone. It in the similar fashion, it activates its receptor, that is RLHR, and in response to that, it activates many elastases. Along with that, it also activates rhamnolipids formation, hydrogen cyanide, and pyocyanin. All these things helps in the adhesion and formation of biofilm, and along with that the host defense mechanism degradation. That means they are helping to increase the virulence mechanism in this bacterium. Similarly, PQA system is also there, that activate by the quinolone signal, and there is a positive feedback mechanism between the RLS and PQS system. Another one is IQS system. It only activate during the stress conditions, for example, phosphate limitation condition. Likewise, so the main objective of my study is to identify some natural compounds which are capable of inhibiting these quorum sensing regulatory proteins, LASER and RLHRs in Pseudomonas aeruginosa by using a multifaceted in silico approach. So the specific objective are to curate the phytocompound library from literature validated antibacterial plant and to screen those compounds by using drug likeness, toxicity, ADMET profiling, and finally to identify some lead compounds against those receptors by using the molecular docking and molecular dynamic simulation method. This is the whole methodology of my work. First, I have selected the natural compounds based on their antibacterial and anti quorum sensing properties and retrieve their 3D structure from the PubChem database. Simultaneously, I have selected the target protein and retrieve their structure from PDB database. Along with that, then I have screened those vast library of natural compound by using different databases like Swiss ADME, ADMET SIR, Protux, and many other databases. And finally, the top virtually screened compound were undergoes for the molecular docking processes by using the Autodoc Vina. And the docked compound I have selected from molecular dynamic simulation method and followed by the post simulation analysis trajectories. My this much I have completed, and my next work is for experimental validation of those compounds. So here is the virtual screening table, including the only two top compound that shows highest affinity towards these two receptor respectively, that is RLHR and laser, prunetin shows highest binding towards the laser, and galangin shows highest binding towards the RLHR. So I have mentioned all their physiological properties along with their toxicity properties here. Here is the molecular docking and interaction analysis. I have shown the binding mechanism and the interaction residues of the compound against the target proteins. So for prunetin, I have docked that compound along with the uh, laser wild type variety and a mutant variety of laser, that is L130F. This is a single point mutation. I have included this for better proposal. Along with that, I have used galangin compound for RLHR. Similarly, I have used the wild structural variety of laser RLHR, and along with that, I have used a single point mutation likewise, that is S135A. So we can see S135A was the active site of that RLHR protein. Hence, after the active site mutation, the binding affinity we can see with galangin along with that compound decreases as we compare to the wild type variety. So these are the two compounds, and in the right hand side I have shown you the interaction of those compound against those proteins. So the green region showing the hydrogen bonding, pink regions are showing the pi-pi interactions, and the orange regions are basically showing the hydrophobic interactions. So here is the post-MD modular dynamic simulation trajectories. The first one is the RMSD, that is root mean square deviation, that shows the total deviation of the protein and ligand complex as compared to the structure that was initially before the molecular dynamic simulation method. And we can see and the first one is for galangin, and galangin shows little bit of fluctuation in the beginning, but later it get stabilized. And for prunetin, it shows throughout stability in the 100 nanosecond simulation period. Next one is the radius of gyration. That shows the compactness of the protein ligand complexes throughout the simulation period. In this, we are seeing how much they are deviate from the center of the mass, and we can see throughout the simulation period they are maintaining the stability. Next one is the RMSF, that is the root mean square fluctuation. In RMSD, we are seeing the whole protein ligand complex fluctuation, but in case of RMSF, we are only seeing the amino acid fluctuation that are present in the wild type and mutant variety of the proteins. Next one is the hydrogen bond. I have mentioned all the four complexes here, and in the y-axis I have mentioned the number of hydrogen bond, in x-axis the simulation time period. So we can see in all the four complexes 2 to 3 hydrogen bonds are continuously forming throughout the simulation period. That means the compound have the affinity to bind with the target proteins. And next one is the dynamic cross correlation matrix. This actually shows the motion of protein and ligand complex. It basically means the red region are showing positive correlation. That means the ligand and proteins are moving in the same direction. And the blue regions generally shows the anti-correlation. That means there is opposite direction of movement between the ligand and protein. So we can see most of the regions are red regions. So we can say the ligand is stably binding to the protein and the mutant variety also. Next one is the principal component analysis and the free
Rupalina?
energy landscape study. Yes, ma'am.
You have only 2 minutes to present, okay?
Okay, ma'am. So principal component analysis shows the motion of the protein in clustered form. So more cluster means more stable, and the FEL shows the global minima state. So we, by the confirmation from the conformational sampling, so we can see the blue region shows the global minima state. That means protein is maintaining its the highest conformation state throughout the simulation period. So next one is the binding free energy analysis that shows all the type of interaction that helping the ligand and protein interaction, including van der Waals force, electrostatic, polar and non-polar separation, and finally the del G total. As the del G total is in the negative side, that means the reaction is feasible and spontaneous. The key findings of this study is I have identified some lead compound against the two target receptor LASAT and RLHR that's with showing better binding affinity and stable interaction. The MD's trajectories also support the stable conformational behavior. The binding free energy calculation also shows a stable and sustainable receptor ligand interaction, and all the advanced analysis I have done, they also supporting a stable and low fluctuation binding conformation. So in conclusion, this work is a systematic in silico screening pipeline that integrates the drug likeness, ADMET, and molecular docking to identify some promising natural inhibitors against the quorum sensing regulatory proteins LASER and RLHR. The molecular dynamic simulation provides structural stability and binding site interactions, and the free energy analysis also refining this study by showing a negative del G, that means the reaction is feasible. And the next step is the in vitro validation by using the biofilm inhibition assay and the quorum sensing inhibition assay. So from this in silico to in vitro strategy, it will provide me some promising candidate for antivirulence development against this multi-drug resistance Pseudomonas aeruginosa bacterium. So these are the references I have used, and thank you.
Yes, thank you, Rupalina. I request our evaluators, Dr. Praveen Kumar Wankhede sir, to have some questions.
Hello, good afternoon.
Good afternoon, sir.
Yes, it's a nice presentation.
Thank you so
So
much,
which
sir.
software you are used for the network pharmacology?
Sir, I did not use the network pharmacology study here.
Okay, directly you done the docking?
Yes, yes, sir.
Yes, okay. So which target you identified?
Sir, I have basically taken the laser receptor that is a quorum sensing regulator of Pseudomonas and activate during the initial exponential phase of this bacterial growth. And along with that, I have taken another target regulator, that is RLHR, that activate in the last stage, that is the late exponential phase of this bacterium. And both these receptors are helping towards the virulence mechanism and biofilm formation in this bacterium.
Which shows the most very good docking score?
Sir, I have seen prunitine is showing minus 10.4 kilocalorie per mole binding affinity towards the laser, and in comparison with that, galangine is showing minus 9.3 binding affinity towards the last RLHR receptor.
Is this P aerogenicide gram positive or gram negative?
It's a gram negative bacterium.
And causes which means which disorder, which disease, or which
It
involved in which disease?
Yes, sir. It is responsible for mostly the respiratory diseases, and it is severe pneumonia also, and it is mostly affecting the immunocompromised individuals and the medical patients also, because it can easily grow in the medical equipments.
Yes, I think it's also involved in the wound healing. Wound.
Yes, yes, sir.
Yes, gram negative. Okay.
Yes, sir.
Okay, so hospital acquired, if there is a hospital acquired infection, so this P regions are involved in it?
Yes, sir. It is totally a hospital-acquired infection, and it is transmitted through that also.
So we have used some animal model for this
No,
for this
sir.
study.
We did not use any animal model. We are just experimentally validating those compounds by taking two bacterial strain. One is the Pseudomonas aeruginosa PA01 strain. Another one is PA14 strain.
Okay.
Yes,
Okay,
sir.
so this study can be possible in animals. So by using this, if you incorporate this P. aeruginosa
Yes, sir.
and
We can
produces some.
also add the in vitro studies also by taking some model organisms.
Okay, okay. So in
In
future you
which
can
we
do it.
can. Yes, sir. I will plan to do that. In that case, we can purify those proteins from this bacterium, and then we can express those protein in the model organisms. So that will be the purification of protein studies.
Okay, yes. Thank you. Thank you. Very good presentation and answer. Over to you, ma'am.
Thank you, sir.
Yes, thank you, Rupalinath.
Thank you so much, ma'am.
Yes.
Can I stop presenting?
Yeah, okay.
Thank you.
So now I request our next presenter, Shalini R,
Ah,
to present your
yes,
work.
ma'am. Yes, actually, my laptop has some problem because I cannot present that. Okay, thank you, thank you. So I'm
Yes,
Dr.
yeah.
Shalini, Assistant Professor, Division of Biotechnology, JS Academy for Education Research, UT Campus, and my topic is computational design and in silico human profiling of multi-epitope vaccine construct targeting major virulence factors of Corynebacterium diphtheriae. Next. So coming to the introduction, respiratory infection disease are remain challenging because due to high mortality, and diphtheria is an acute respiratory disease which is usually toxin mediated disease caused by Corynebacterium diphtheriae. In severe cases, as we can see the cardiac and neurological complications also. So, but current therapy mainly involves using the antitoxin, antibiotics, and toxoid vaccine. So mainly antibiotics will which lead to the antibiotic resistant, and antitoxin which is available DAT, that is diphtheria antitoxin, which is having a poor affinity with the antibody. And also you have another one more toxin which is available in the market, that is DPT, that is diphtheria pertussis tetanosus, that is also available in the market, but it is a combination vaccine because different antigen can be incorporated and it has been given, so it will be having a safety concern. And it should have a say that because of that you need to develop so effective treatment will be needed to increase the cell mediated as well as humoral immunity, and with a high population coverage and with a long lasting memory cells. So it is necessary to develop a multi epitope vaccine that can able to enhance both humoral as well as the cell mediated immunity with a higher population coverage with long lasting immunological memory, because DPT which is having a lower population coverage, because of that reason we have to increase the population coverages also. Next slide please. Next slide. Okay, based on that my aim is to develop the conserved multi-antigenic epitope to construct a safe, stable, and immunological multi-epitope vaccine candidate against Corynebacterium diphtheriae using reverse vaccinology and immunoinformatics techniques. So, based upon the aim, my objectives are to identify conserved antigenic protein of Corynebacterium diphtheriae for vaccine development, to predict and screen the B cell epitope, T cell epitope, as well as the helper T cell epitopes using immunoinformatics tools, to evaluate the selected epitope for antigenicity, allergenicity, toxicity, and immunogenicity, and to construct the multi-epitope vaccine candidate by using adjuvant as well as the linker, to predict 3D and 2D and 3D structure, and able to validate the structure of the 3D protein, and to investigate vaccine receptor interaction through the molecular docking and molecular simulation process. And we have a codon optimization and in silico cloning also. So my methodology, the protein have a series of steps. Protein sequence are retrieval. So we have utilized uniprot kb, and to find out the antigenicity and allergenicity and toxicity prediction, we have utilized vaccine, allertrop, and toxin prediction. And epitope prediction, we have utilized IEDB. And for the multi-epitope vaccine construct, we utilize different adjuvants as well as the linkers. And to the population coverage, we have utilized the IEDB population coverage tool, and we have found out the physicochemical properties, solubility of the particular multi-epitope vaccine, and secondary and tertiary structures were predicted and refined and validated. And we have done the molecular docking and molecular simulation studies, and also we have done the immune simulation study by using C immune sim, and also we have done the codon optimization and in silico cloning. And coming to the resultant discussion, so these are the three protein which has been taken from the uniprot, because diphtheria toxin protein and fimbrial subunit protein and membrane protein these are the major cause for the diphtheria vaccine, diphtheria disease. So because of that reason, we have taken three different proteins, and the three different proteins also we have evaluated with antigenicity, allergenicity, as well as the toxicity by using different tools. And the threshold value is 0.4. If it is above 0.4, it is as considered as antigenic, so we can take that for the further process. And these are the protein we have retrieved from the uniprot database. And next these are the protein we have retrieved from the uniprot. Then next. Okay, so these are the MHC class 1 molecule epitopes. So we have taken three different protein sequences. From that, we have found out the MHC1 class 1 epitope, and we also found out whether it is antigen, allergen, or toxicity production. So based upon that, we have kept it as a 9 amino acid sequence for class 1 MHC, because it will be having a higher binding affinity with the MHC class 1 epitopes. Then next. Okay, so this is the MHC2 molecule. So for this, we have taken three protein, and we have predicted MHC class 2 epitopes also. So whichever is non-antigenic, whichever is non-allergen, whichever is non-toxic, that can be further taken. So here I've given the selected epitopes which is having higher binding affinity with MHC class 2 epitope. Next. So next one is B cell epitope. From these, we have found out the B cell epitopes also, and also the B cell epitopes also, which has been predicted with antigenicity, allergenicity, and toxicity. And we have selected some epitopes which is having higher binding affinity with the B cell epitopes. Next. So, and also we have found out the population coverage for the MHC1 and MHC2 separately, and we have found that is 61.5 percentage of for MHC1 and 36.86 percentage of coverage for the MHC2. When we combine both, we have got the population coverage of 75.69 percentage of population coverage. So it is a good population coverage, because when you combine MHC1 and MHC2, it will be acting as a 75 percentage of the whole population coverage has been covered. And multi-epitope vaccine construct, we utilized various next. So we have utilized various linkers, adapter, and also the adjuvants to increase the efficacy of the vaccine. So here we have utilized a paired epitope and RS09 adjuvant for constructing the multi-epitope. So here, whatever the sequence which has been represented in the red, that is known as the MHC1 molecule, that has been connected with AAY linker. And whatever that is displayed in the green color, that is a T cell epitope that has been connected with a GPGPG linker. And the blue color, that is purple color, that is a B cell epitope that has been connected with KK linker. And we have designed two different vaccines. Okay, one with the adjuvant, that is paired adjuvant, and other one with adjuvant, that is RS09 adjuvant. Because why we have selected these two adjuvants? Because paired A will be having a more affinity towards the TLR proteins, TLR receptor, and also you have RS09, which will have a higher affinity towards the T cell receptor. So because of that reason, we have taken two different adjuvant sequence to construct the multi-epitope vaccine. Next. And when coming to the allergenicity, so it is the whole construction was evaluated for the antigenicity, allergenicity, and toxicity. And we have both are having good antigenicity. Both are non and allergenic and also the non toxic. Based upon that, we have taken, and we have analyzed the physicochemical properties also. Mainly we have concentrated on the solubility, because it is 85 to 85 percentage solubility, where the both the proteins have, both the epitopes will be having a 85 percentage solubility. And after the construction of the vaccine, we have converted, we have identified the secondary and tertiary structure also. So here we can see the next.
Shalini ma'am, you only have 2 minutes more to
Okay,
present,
okay,
okay?
okay.
Yes.
Next. Okay, so and also we have found out the physicochemical property. The solubility is major thing. So 85 percentage of solubility was there for to both the construct also. Next. Okay, we have predicted the secondary and tertiary structure. Next. And both and the every both the epitopes also will be having a pole polarity, that is overall clash and overall the efficiency and lowest clash score, everything is a limit, everything is optimal. So because of that, we have taken both. And next. And we have done the docking studies also. In the docking studies, we have found that it is the RSO post 4 will be having a higher binding affinity, and here RS pad A sequence will be having post 4 is also having higher affinity. So because of that, it is having a higher binding affinity with the TLR4 protein, because we have taken a TLR4 protein because it is the main thing for the innate immune system. So we have taken that when we analyze the docking score also. Then next. Okay, so then we have found out, we have done the molecular dynamic study also. In that, protein ligand has the good affinity. So when compared to the part A, the RS09 will be having more affinity towards the protein, and that can be mainly, it will be connected with the tyrosine 36. That is a major connection here, and also most of the connection will be a hydrophobic connection here we can found. Next. And we also done the immune simulation by using C immune. So here mainly it will be producing the B cell as well as the interleukin 2. So B cell mainly you have B cell stimulate that only the IgM and IgG. So both will be having a higher concentration, and also the antibody titer also higher, and interleukin 2 response also higher. The interleukin 2 which will be responsible for activating the T cell, and it will enhance the proliferation. So it will be having a good immune simulation. So when you are injecting into the human beings also, will be having a good effect. And we have done the next. And we have now done the codon optimization to express well in the host system, that is in the E. coli. So because of that reason, codon optimization, after we have done the codon optimization, we have seen the GC content also in first that pad A adjuvant. When we handed the pad A adjuvant, that has a 52.38 GC content, and next RS09 have the 53.29 GC content. So it will be giving a good protein expression, that we can conclude that. And next, we have done the in silico cloning also. So whatever the protein we have taken, and that can be converted into DNA sequence, and we have done the in silico cloning also by using the PET vector, that is expression vector. So this can be mainly, it can be included in the E. coli. So once if it is expressed in the E. coli, then we can take out that, and we can do the we can purify the thing, and we can utilize for the human beings. And finally, conclusion. So conserved epitope vaccine we have constructed the with the help of the T cell epitope, B cell epitope, as well as the cytotoxic T cell epitope, helper T cell epitope, and also the B cell epitope. So all the epitopes were non allergic, non toxicity, and it has having a favorable antigenicity, and it can be confirmed we have conformed with the structure, that is we have done the 3D structure as well as the 2D structure, and we have done the molecular docking also, then it has a good affinity. So when compared to the part A, RS09 will be having a higher affinity towards the TLR receptors, and here the multi-epitope vaccine is a good candidate for to treat the Corynebacterium diphtheriae, and further in vivo in vitro studies to be done. So this is the part of the studies which have done in the presented here. So these are my references, and thank you.
Thank you, Shalini ma'am. We will have
Thank
one
you.
question for you.
Oh,
I
okay,
request
tell
our
me.
evaluator to have one question to Shalini ma'am.
Yes.
Thank you, Dr. Shalini.
Thank you, sir.
So,
Sir,
what
and
was
also I'm because my slide was not shared. I mean, I had a little problem, so I was tensed. So
no,
sorry,
no,
sir,
it's
for that.
fine.
And
That's
thank
fine. So,
you.
what was your, the what gap actually you are trying to solve here?
Sir, actually, the whatever the vaccine we have, that is a low population coverage, because DBT it is a combination of vaccine. So we have three different antigen which has been combined, and that has been given to the patient. So, and also that is having a higher, that is safer. You have to see the safer concern, because each and every time when you see the switching of the antibody will be differ when you have all the three antigens. So what we thought, we construct the multi epitope. Okay, so that will have a higher it can able to enhance both humoral as well as the cell mediated immunity. And also we can see that the higher population coverage, because whatever the vaccine we have now, that is not a higher population coverage, and it is frequent boosters are needed. So to overcome all these things, we have done the multi-epitope vaccine, because here we have a T cell epitopes, B cell epitopes. As in the T cell epitopes also, you have a cytotoxic T cell as well as the helper T cell. So when you're combining that, you can able to enhance the activity of the particular vaccine. That is what my idea, sir.
Okay, thank you.
Thank you, sir.
Thank you,
Thank you.
Shalini ma'am. Yes, thank you, ma'am.
Thank you, ma'am. Thank you for the opportunity. Sir, if I want to become a member of a Swalife, what can I do for this?
You can contact just contact the same numbers which are there as a group admin, and we
Okay,
will
okay.
yeah. So we will get back to you this one.
Okay, sir. Thank you. Thank you.
Thank
Thank
you.
you, sir.
Thanks.
Thank you. Okay, sir.
Yes, now we will invite our next presenter. So our next presenter is Hazel
Sorry,
Rins.
ma'am.
I request Hazel Rins to present your work. Uh, am I audible to you, Hazel Rins? Hello, am I audible?
Yes, you're audible.
Okay. Hazel Rins, can you hear me? Hello, Hazel Rins. Oh, Hazel Rins, we can't hear you. Hello, Hazel Rins, we can't hear you.
Hello, Hazel, can you hear us? Ruchira, I think there is a issue with the audio. Can you take next person?
Yes, sir. I request Prathamesh Gudgila to present your work. Hello, Prathamesh Gudgila, can you hear me?
Yes, ma'am.
Okay, share your presentation, please.
Yes, ma'am. It is uploading now.
Okay. Okay, Prathamesh, you will have 10 minutes to present your work and your time starts now.
So, good afternoon, everyone. My name is Prathamesh Gudgila, and I am pursuing Master of Pharmacy from Vishwakarma University. And my topic name is Preclinical Evaluation of Antioxidants for Potential Oral Cancer Chemoprevention. In that, my combined antioxidant extracts are rosmarinic acid, ursolic acid, and methylphenidate. So, introduction. Oral cancer is an important health problem worldwide, in particularly in the India. Approximately 90% of oral cancer are of oral squamous cell carcinoma, and its major risk factors are like tobacco use and smoking, and betel quid chewing, and HPV infection. So the major problem of oral cancer is that it remains asymptomatic during the early stages, so its diagnosis may be delayed. In this figure, you can see the various affected areas of cancer, including lips, buccal, mucosa, tongue, floor of mouth. So, therefore, early detection is very important. So, we need to investigate safe and effective preventive or chemo preventive approaches for this. So, these are the global and Indian burden of oral cancer. In this, oral cancer is the sixth most common cancer worldwide. More than 3,77,000 cases are there every year. So nearly 30% of global oral cancer burden according this information. In this, the 2 to 3 times higher are of males between the ages of 40 to 70 years. So tobacco and alcohol are the major contributing factors. However, early detection can significantly improve these outcomes. In this, it is important for risk factors shown here, such as these are tobacco, alcohol, yes, betel quid, and HPV infection. So among these, tobacco and alcohol and betel quid are particularly important and modifiable risk factors. So repeated exposure of this using can cause the oral cancer. So these changes can be contributed to abnormal cellular growth and eventually malignant transformation. In this slide, the pathophysiology of oral cancer. Oral cancer develops through the accumulation of several molecular and cellular changes. In this, molecular changes are like DNA mutation, oxidative stress, cell cycle dysregulation, reduced apoptosis, and angiogenesis, and invasion and metastasis. Normally, cells have the mechanism that control their growth and remove damaged cells through the apoptosis. But repeated molecular alteration can then lead to the malignant transformation. So this is the important for the study that oxidative stress is the one of the mechanism associated with for the cellular damage and carcinogenesis. In this oxidative stress, exposure to the carcinogens such as tobacco use, alcohols, can generate the reactive oxygen species. But excessive ROS, that is reactive oxygen species, can damage important cellular components. So it can cause damage to DNA damage, lipid peroxidation, protein damage. So which may contribute to the mutation and genetic instabilities. So these changes can support the process and involve in the cancer development. So these antioxidants can use such as rosmarinic acid, ursolic, and vitaferin A. Because of their reported antioxidant activity and biological property, they can scavenge this reactive oxygen species. The aim and objective of this is that we want to evaluate the main preliminary clinical effects of combined antioxidant extract, in which containing the antioxidants. Our first aim is to that we prepare the combined antioxidant extract. Then we will observe the appropriate control and standard groups. Then antioxidant activity was evaluated using the DPPH scavenging assay, and preliminary toxicity was evaluated using an earthworm model. So together, these experiments were intended to provide initial biological evidence for further investigations. Need and rationale. The rationale for this study comes from the significant burden of oral cancer and need for additional preventive approaches. Conventional cancer treatment can be associated with the limitants such as adverse effect and high treatment cost. So therefore, natural antioxidants are used. And since oxidative stress is associated with cellular damage and carcinogens, so antioxidant activity is the step which can be used. So therefore, combined antioxidant extract and for antioxidant activity is important. These are the selected antioxidants. In this rosmarinic acid, it is a polyphenolic compounds which is having a strong antioxidant activity and anti-inflammatory activity, which can scavenge the free radical scavenging activities. Its sources are from rosemary and oncinium. Ursolic acid is the pentacyclic triterpenoid with reported having the antioxidant activity and anti-inflammatory activity. And third one is the vitaferin A, which is commonly known as ashwagandha. It has been investigated for antioxidant and anti-inflammatory and also the anti-cancer related activity. So these are the three compounds were selected for the for based on their reported biological properties. So this assay was performed DPPH antioxidant activity. In this, DPPH is a stable free radicals that produces the violet color. The antioxidant reacts with the DPPH. The radical is then reduced and the color intensity decreases. In this, the reduction of absorbance can determine the radical scavenging activity of the sample. So in this, we uses the antioxidant extract. So in this, we use 1 ml of drug sample and 1 ml of DPPH. Then we kept in for 30 minutes in dark, and we check its at absorbance at 520 nanometer. So it is the graph which shows the activity. The standard one is the ascorbic acid. So this come out of the assay. This shows that the combined antioxidant demonstrate the significant DPPS radical scavenging activity. So next we perform the earthworm toxicity assay. In this we perform this for evaluation the preliminary toxicity of combined antioxidant extract using an earthworm model. In this, the control group of earthworm was maintained in a moist soil. Then the antioxidant extract was treated at the concentration of 1 mg per ml was taken and treated to the earthworm. Then we have observed the earthworm at 48 hours, 7 days and 14 days. And we checked their parameters like the survival and movement activity. Then by comparing it with the control group, we determine the safety of its earthworm. So in this control, up to 48 hours, three were alive, and then on seventh day, one earthworm was dead of the control, but of the extract we use antioxidant was not dead. So by this we give the ratings. These are the pictures which I have taken for the earthworms. We can see their movements, and which I have given the rating for 4: active movement, normal behavior. So next study I was done is the antimitotic study. The antimitotic study was done for checking the regeneration of earthworms. Earthworms were cutted from the 13 segments from the then after this we have taken the earthworms and checked up to the 7 days on third, fifth, and 7 days, and then put the sample of antioxidant extract at the various concentration. So these were the results. On we uses concentration like 1 mg per ml, 0.5 mg per ml, and 0.250 mg, 0.125 mg per ml. In this growth were of control group on third 25% and fifth day 50%. So, so these are the photos of antimitotic study in which we can see that the control on the 7th day it has got the 75% growth. So this study was performed, and with this we can say, compare with the control and said that the regeneration of antioxidant extract we use has less. So this is the conclusion that the combined antioxidant extract containing these antioxidants can measurable DPPH scavenging activity, and the extract we use have the 100% survival rate in toxicity rate up to 14 days we have checked. Then on the regeneration we can show it has the reduced regeneration compared with the control. So further studies we can establish for the mechanism and for this. So these are the references which are used for making the PPT. Thank you.
Okay, thank you, Prathamesh. I request our evaluator, Pawan Kumar sir, to have some questions. Hello, am I audible to you, Pawan Kumar sir? Okay, I request Dr. Praveen Vade sir to have some questions.
Thank you for the presentation, Prathamesh. So in the toxicity study, so you used earthworm. So in the antimitotic also, did you use earthworm? How was the antimitotic assay performed?
Yes, sir. For antimitotic also we have used earthworm, and we have
Okay.
observed on the third, fifth, and 7th day.
Okay. So what actually you are trying to solve? What is the problem? What is the gap? Hypothesis.
Sir, the gap is that we have to see the antioxidant and anti-cancer activity of this drug. So that's why we perform this test, DPPH and earthworm toxicity. So we can check that the antioxidant activity of this all these extracts which have used.
Okay. Thank you, Prathamesh.
Thank you, sir.
Okay, thank you, Prathamesh. Now I request our next presenter, Hazel Rings, to present your work.
Good afternoon. Am I audible? Am I audible?
Hello? Yes,
Hello,
you are
yes. Am
audible.
I audible? Okay.
Yes, you are audible.
Good afternoon, respected sir and ma'am. My name is Hazel Rinja. I'm a research scholar from Department of Optometry from Royal Global University. And today I'm going to present about the narrative-free view, that is the clinical assessment and management of sensory and motor sense fusion, a narrative review of binocular vision and outcome. So this is the presentation outline. Introduction. Binocular vision is the ability of both eyes to work together and to coordinate to form a single clear image. It depends on the ocular alignment, the retinal image, and the neural pathway. If there is any problem in the sensory or in the motor fusion, the patient may experience a symptom like diplopia or a double vision, suppression, eye strain, a headache, and a reduced stereopsis. Because mainly the brain, it will combine the information from both eye to produce a stereopsis or the depth of perception. As you can see here from the figure, this is the figure where it illustrates of how binocular vision is achieved. The light, first of all, it enters into the retina, then it passes through the optic nerve, to the optic chashma, to the optic tract, then to the lateral geniculate body. After that, the signal it will send to the visual cortex or the brain to form as a single image or a clear image. And as you can see it here, there is a sensory fusion and a motor fusion. So mainly sensory fusion is to combine the two retinal image to form into a single image. So mainly it prevents the suppression and the diplopia or the double vision. And the motor fusion here, it maintains the ocular alignment or the eye movement. So it will keep the image on a corresponding retinal image. So it can make it clear and easily to see the depth of perception. And the next is the rationale here. The binocular vision disorder are commonly encountered in optometric practice, but most of the time we overlook during the routine eyes checkup. So the early diagnosis, it allows the intervention before the symptom it become more severe or bad. And numerous clinical tests are available to assess the sensory and motor fusion, and recent development in office-based vision therapy or a digital rehabilitation and some orthoptic training, it really improved for the treatment or the management outcome for those patients who have anomalies in motor and sensory fusion. So the main aim here is to review to evaluate the current evidence regarding the clinical assessment and the management of sensory and motor fusion and the role to improve the binocular vision outcome. And the objectives are, the first is to explain the physiological mechanism of sensory and motor fusion. The second is to review the commonly used clinical assessment technique. And the third one is to discuss the evidence-based management strategies and the use in a binocular vision rehabilitation. Methodology here conducted as a narrative review, and the relevant literature were obtained from the PubMed, the Scopus, Google Scholars, and Science Direct. So the main keywords I used that review articles for motor fusion, sensory fusion, binocular vision therapy, orthoptic, and divergence disorder. So for the clinical assessment here, we begin with the evaluating first the sensory fusion, then the motor fusion. So first, when it came about the sensory fusion, the assessment R's, Watford dot test is a test to detect the suppression and diplopia. The second one is the Bangolini straighted glasses, where it assesses the normal binocular sensory. And Synoptophore is the instrument where we use to check both the sensory and motor fusion. And it also helps for the objective and the objective of the angle of deviation. So the last one is the stereopsis, which is helpful for the depth of perception. Suppression testing, mainly we use if one eye is being ignored by the brain, so it can also lead to diplopia. So motor fusion assessment, these are some tests, the cover test, to identify if there is any ocular deviation or alignment. Prism bar vergence testing to measure the fusional vergence amplitude, near point of convergence to assess the convergence ability, and the vergence facility to evaluate the flexibility of the vergence response. And the clinical management, there are some treatment where we can manage it, but it also depends on the underlying causes. So the first one is correction of refraction, mainly if the causes is due to the refractive error. And prism therapy, it also prescribed to reduce the diplopia or double vision and to relieve the symptom. And vision training is also giving to reduce the management, to reduce some like diplopia, and office-based vision therapy to exercise or to improve divergence, accommodation, and the binocular coordination. A home-based therapy, mainly we used to give, this is just a kind of exercise, orthoptic training to strengthen the fusional vergence and to reserve the ocular alignment. Digital vision therapy, this is mainly the software and some virtual reality and AI. And the last one is the patient education and regular follow-up. It is important to monitoring the progress and the improving of the treatment. So the clinical implication here, the finding of this review have several clinical implications. That is the comprehensive assessment to allow the early diagnosis of binocular vision disorder and management. It improves the binocular visual function and the patient comfort. Successful treatment, it enhances the quality of life and the academic performance or the occupational efficiency and sport performance. So my conclusion is that sensory and motor fusion are the fundamental components of binocular vision, and a comprehensive clinical assessment of both sensory and motor is essential for the accurate eye diagnosis and the evidence-based management, including refractive correction, prism therapy, orthoptic exercise, and vision therapy. It really helps to improve the binocular visual function, and these are some of the reference, and that's all. Thank you so much for giving me this opportunity.
Okay, thank you, Hazel Range. Now we will have some questions. I request Pawan Kumar Wankhede sir to have one question to Hazel Range.
Yes. Can you summarize or conclude your presentation in 2 minutes?
Yes, sir. The main is that there are some therapy that is for those patients who have sensory or motor fusion problem anomalies. They will have the problem like they will always complain of eye strain, headache, and double vision. So in this case, there are some treatment like we cannot do surgery. So in that case, we are giving some therapy. That is, if it is due to refractive error, that is due to myopia, hypermetropia, or astigmatism, um, we used to give this correction first of that refractive error, and then after that we used to give this prism therapy if there is any ocular alignment, that is the motor fusion, and vision therapy. Office based vision therapy, we give some exercise to the patient to exercise due to any accommodation problem, vergence problem. And home based therapy, mainly we used to give for, mainly nowadays what happened after COVID-19, we most of the kids are having myopia and they are having some sensory and motor fusion problem. So we used to give a home based therapy as an exercise for them, and we will advise them to play outdoor more instead of spending more time in the screen. And some digital vision therapy we used, that is computer software, and some virtual reality we used to provide. And the patient education is for mainly for this progress. We have to check how much like after the follow up we have to check whether this sensory and motor assessment, it is still increasing or it is decreased or it is in the normal stage.
Okay. Can we develop some animal models or in vitro or in vivo models to validate this?
Yes, sir. Yes, sir. We can develop that. We can do for that, like in the form of patent, we can develop some instrument. But the first one is that what I'm here in my review article is I'm just discussing about some assessment, like where we can access, because mainly we used to ignore this binocular assessment. It is difficult, or it is totally different with the comprehensive eye examination. So we used to do in a separate way. So that is the reason, like if any patient have this problem, so we used to do this assessment, and then we will give some therapy.
Okay. Okay. Thank you. Over to you, ma'am.
Thank you, sir.
Yes, thank you, ma'am.
Okay, thank you, ma'am.
Yes. Thank you to all our presenters, and we sincerely thank to all our evaluators for their valuable contribution to this conference. With this, we have successfully concluded all the scientific sessions scheduled for day 2 of the Swalife Biotech 4th Annual International Conference 2026. We shall now move towards to our final keynote lecture. It is my privilege to introduce our international speaker, Dr. Elena Chitti. Dr. Elena Chitti is an accomplished molecular biologist whose research focuses on understanding the molecular mechanisms and regulate gene expression in bacterial pathogens, particularly Helicobacter pylori, Campylobacter genuine. During her doctoral research, she investigated essential transcriptional regulatory using molecular biology, microbiology, and functional genomics approaches. Her current research interests include bacterial transcriptional regulation, host adaptation, and identification of novel antimicrobial targets, contributing to deeper understanding bacterial pathogenesis and future therapeutic strategies. Today's keynote lecture is titled Unraveling Chromatin Dynamics and Regulatory Networks in Epsilon Proteobacteria. This session promises to provide insights into microbial genetics and modern molecular biology. Everyone, please join me in welcoming Dr. Elena Chitti. Ma'am, this stage is all yours.
Thank you. Can you hear me?
Oh, yes, ma'am, we can hear you.
Okay. I can now try to present.
Yes, ma'am.
Oh, well. I don't know. Okay. Maybe you can see now the presentation.
Yes, ma'am, we can see.
Okay, perfect. Thank you for the nice introduction and thank you for having me. Today I will talk about my PhD project about regulatory networks in Epsilon proteobacteria. I will not expose all my results, but I want to make you understand how study molecular processes in bacteria can help us pave the way for other studies and to identify new antimicrobial targets. So first here you see the outline of this presentation, so you can help, this can help you following me during this talk. First of all, I will just give an introduction on why do we study bacteria and how we can identify new antimicrobial targets. Then I will move on to the first project about Campylobacter jejuni, and then move on to the second one on Helicobacter pylori. And at the end, some take-home messages and future perspectives of these projects. So first of all, why do we study bacteria? Bacteria are ubiquitous, so they can colonize almost all environment, also the human body, in which they can have positive a role, for example, the microbiota, they represent also a public health threat because they can lead to several disorders and pathologies. So it's important to try to understand their physiology in order to prevent these pathologies and problems, and also because there is an increasing antimicrobial resistance from bacteria, so there is an increasing interest in trying to find new therapies against them. And finally, they are also a very interesting model to try to study all the molecular processes and physiology in science. So why do we study them? Because try to understand molecular processes and adaptation from bacteria through the environment or stresses can help us understand how we can fight them. And this is the basis for the identification of new antimicrobial targets, because by understanding the bacterial biology, we can identify some processes, and some of them are essential processes. So if we block them, we can prevent the growth of the bacterium. Inside of this mechanism, there are several actors and proteins that can be exploited to study and identify new compounds that can have an antimicrobial effect. And the main purpose of my project was this one. So try to deepen the knowledge that we have on two pathogenic bacteria, Helicobacter pylori and Campylobacter jejuni, in order to pave the way for other studies and future studies to develop other therapies against these two pathogens. Just to put my project into a context, here you see a multi-layer regulation of gene expression in bacteria. We know that this can happen at different levels. At the most local level, with some post-translational regulation and modification of the protein, for example, with phosphorylation or redox switches that can change their function and structure. Through a more broader scale with the control of the three-dimensional structure of the genome through supercoiling or interaction with proteins. And this can lead to the control and changing in a lot of gene inside the bacterium itself. And my two projects assess the two first level of this scheme, because in the first project I characterized the regulon of a transcriptional factor in Campylobacter jejuni. This transcriptional factor is called COS-R, while in the second project I developed a novel technology to study the three-dimensional structure of Helicobacter pylori nucleoid. So I just wanted to, just to follow me better, now we can move to the first project talking about this bacterium that is Campylobacter jejuni. This is defined as a foodborne pathogen because it normally resides in the gastrointestinal tracts of poultry, sheep, and cattle without causing symptoms, and can infect the human host through contaminated food and water, generating foodborne gastroenteritis, but also neurological disorders such as the Guillain-Barré syndrome. As I said, there is an increasing resistance in bacteria and also for Campylobacter jejuni against the current use therapies. So there is a new field to try to identify the new antimicrobial targets. And in this scenario, one of the most promising target is represented by COSAR. As you see, this is a regulator, a transcriptional factor, defined as an essential transcriptional factor of Campylobacter, because we cannot generate a knockout mutant of this protein because the bacterium is not vital. And it seems to be involved in many processes such as metabolism, macromolecule biosynthesis, and energy production. And it has also been defined as an oxidative stress response regulator that is very important for Campylobacter because it is the mechanism that can help him through the zoonotic transition from the animal to human host. In bacterial research, transcriptional factors are emerging as antimicrobial targets because they have a central role generally in bacterium, but they are also specific for the bacterial organism. They have no human counterpart. So we wanted to study better this regulator to identify its function and regulon, because despite all the information that we have about it, all the experiments that are present in literature are based on a PNA technique. PNA are antisense oligonucleotide that can be used against the mRNA of a gene, of a protein. And they can lead to indirect effects. So we don't know if all the information that we have are directly correlated through the function of COSAR or if there are other actors involved in all these processes and mechanisms. This is why the research objective of this first project was trying to use a ChIP-seq approach, so a genome-wide approach to characterize COSAR regulon. So here you see the pipeline, the experimental pipeline of the ChIP-seq. We generate a flag tag strain. So we inserted the tag at the C-terminal region of the coding sequence of COSAR. And then we perform the immunoprecipitation, recovering the protein and DNA complexes after cross-linking. All the DNA fragments were then sequenced and aligned to the COSAR genome in order to identify the genomic position and their biological function. So you see here the result that we were able to perform the ChIP-seq protocol and identified 168 COSAR binding sites, most of them located in promoter regions of genes that are involved in a lot of different mechanisms, for example, translation, ribosomal structure, but also energy production or signal transduction. So this confirming COSAR as a global regulator, and so an essential protein. We then move to the validation of these results through in vitro and in vivo experiment, because on the right we have the quantitative PCR after the chip, and you see the evaluation of the enrichment of 4 targets that we selected from the chip peak profile that we obtained in the flag strain in black, compared to the wild type that was used as a control, because as I said, we could not generate a knockout mutant of this protein. So we used the wild type strain as a negative control. You see that there is an enrichment of these 4 targets in the flag strain, and there is no enrichment of the 16S, that is another gene used as a negative control and that is not a target of COSAR. So this means that our chip was successful and that we have a precise enrichment due to the interaction of the protein with these DNA targets. Also, in vitro analysis through electrophoretic mobility shift assays, so in which we put the protein and the DNA fragments corresponding to the promoter regions of these genes together, show that the protein is able to bind all these 4 targets, because as you can see, we have a shift, so the interaction of the protein with the promoter region, without the interaction through the specific probe. And this binding is present also at a very low quantity of protein. So COSAR has different affinities for these targets, but in any case, it can bind the DNA and confirming the interaction of the protein with this DNA sequencing. But as I said before, COSAR was defined also as a redox protein. So we wanted to see if in our conditions we had a changing in COSAR function and binding on the DNA after an oxidative stress of the Campylobacter culture. So we perform again the ChIP-seq protocol after introducing a redox stress in Campylobacter. And you see on the left here the DNA enrichment levels of the 4 targets that I showed you before, and other 2 that we wanted to analyze, because COSAR is the coding sequence of the regulator. This because in bacteria it's very common that a transcription factor can auto-regulate themselves, so bind its own coding sequence. And AHPC that was identified as a redox target from other experiments in literature. And we wanted to see if this was a real target in our conditions, experimental conditions. If we analyze the DNA enrichment levels of these targets, we can see that most of them have an enrichment in the binding of the protein on the DNA. Different is for RPSF, in which we have a reduction of it. And the interesting thing is that if we compare the DNA enrichment level with the mRNA, so the transcription of these genes, we can see that all of them have a reduction in mRNA level, so in the transcription. Also for RPSF, while for AHPC in our condition we didn't see any changing. So for us is not in line with literature results. But what we can say from these results is that COSAR is sensible to redox stress, because its binding and function change after introducing an oxidative stress, and that maybe it can have a doable role, because for RPSF we have a reduction in the binding but also reduction in the expression, while for the other targets is in higher binding of the protein corresponding to reduction in expression. So maybe COSAR can act both as activator and regulator of transcription of these genes. We cannot directly say that, but we can assume that maybe it can have a doable role. In any case, we suggesting and we are in line with the idea that COSAR is a redox sensitive regulator. I also wanted to report it here other in vitro analysis that we did. There are footprinting assays, so to locate the precise binding of the protein on the promoter regions of the target. And in all of them, as you see, the binding region in blue is near the peak of the ChIP-seq or encompassing the peak. So this is another confirmation of our experimental conditions. And the most important thing is that even if all these targets have a different score because they correspond to different peak in the ChIP-seq profile, they are all bounded by COSAR. And in these three other example, it is just another other figures to show you that COSAR has a global role because it can bind a ribosomal RNA, but also its own promoter, and also a gene encoding for an hydrogenase, so involved in redox response. And so to conclude this first project, what I show you is that we were able to perform a genome-wide characterization of the COSAR regulon through a ChIP-seq approach. We identified in this way 168 high-confidence binding site, promoter regions of genes involved in translation, energy metabolism, and other mechanisms indicating a global regulatory role of this transcription factor that is also sensitive to redox stresses. And so confirming again that this protein is very important and can be evaluable antimicrobial target against Campylobacter. We can now move to the second project, talking about another bacterium, Helicobacter pylori, that maybe you have heard about it because it's one of the most common human pathogens, as you see from this map, with a prevalence of around 20-50% in developed country and reaching 80% in developing countries. So it's very, very common. It is a gram-negative bacterium, microaerophilic bacterium, whose ecological niche is the gastric mucosa. And its persistent infection can lead to several gastric disorders such as peptic ulcer, gastroenteritis, but also adenocarcinoma and multilymphoma. But besides all this interesting information about the bacterium, it is it has a physiological adaptation that is very interesting, because in the viable and culturable state, Helicobacter is a spiralized bacterium, as you see from this image. But under suboptimal environmental conditions, such as starvation, pH and temperature changes, and also antimicrobial pressure, it can undergo a physiological transition to a so-called viable non-culturable state, shortly VBNC, in which the cells become round cells. In fact, these are called coccoid. The VBNC state is common to different kind of bacteria, so it's not only for Helicobacter, and they represent a reservoir of pathogens because they are more resistant, but they are still alive because they maintain intact cell membranes, lower metabolic activity, gene expression. So the interest in try to study this state is to understand the mechanisms that are involved in the physiological transition in order to prevent this state. Also because, for example, for Helicobacter, highly pathogenic strains of this bacterium seems to be more prone to shift toward the VBNC state. So to relapse also to a pathogenic state and generating diseases in the human host. The fact that these cells are alive and that they still have metabolic activity and gene expressions, suggesting that maybe there is a very controlled mechanism that can regulate the VBNC state. And so we hypothesize that can be a chromosome remodeling that can change the expression of a lot of genes inside of Helicobacter. And we know that in bacteria, the genome is structurized by the interaction with the nucleoid-associated proteins, by the formation of chromatin domains, so the supercoiling, and also by epigenetic modification, such as methylation. And despite the small size of Helicobacter pylori genome that is 1.7 megabase pair, it possesses the highest number of restriction modification systems among all bacteria. So this indicates that maybe it has a very high methylome that can regulate the three-dimensional structure of the nucleoid, and it also encodes for some nucleoid-associated proteins such as HOOP or HP119. But in these processes, there can also be involved other actors, for example, HP1043, that is a transcription regulator that is also an homolog of Cosarin Campylobacter, because these two bacterium are phylogenetically correlated. In fact, they belong to the same class, the Epsilon Proteobacteria class. And why HP1043? Because in our laboratory, we perform a knockdown experiment using oligonucleotide, antisense oligonucleotide against HP1043 mRNA, and we saw the generation of an heterogeneous population composed by coccoid cells and spiralized cells, maybe indicating an important function of HP1043 in the regulation of this morphological state. But the nucleoid architecture of Helicobacter is poorly characterized. And in this project, we wanted to move forward to the classical techniques that can be used to study it, for example, the high-C, but also the ChIP-seq itself, because all these techniques are based on chemical treatments that can introduce some artifacts, and we wanted to try to study the H. pylori nucleoid in the native state and understand if it is dynamically regulated through H. pylori growth and specifically in the coccoid state. So in this second project, we developed a novel technology to study the nucleoid architecture, and this technique is called 4F semisec. 4F because it can fractionate the DNA in 4 different fractions based on the accessibility of the DNA itself. In the first fraction S1, we recover the soluble protein. In the S2, the most accessible part of the DNA. In the S3 and S4, the most inaccessible and compacted region of the DNA. This is because all the treatment is based on a DNA's cutting of the genome in the accessible part of the DNA. So the first thing that we have to do is to permeabilize the cell in order to allow the DNAs to enter inside of it and cut the DNA. And then with high salt and urea denaturation, we recover all the other compacted and inaccessible region of the nucleoid. Here you see the schematic representation of the protocol on eukaryotic cells, because this technology was first developed on eukaryotic system and was successfully used on different kind of eukaryotic cells. So the first thing that we had to do was to introduce some adjustment for the prokaryotic system. So the introduction, for example, of a lysozyme passage at the beginning to destroy the cell wall, and then define a precise concentration of the detergent in order to only destabilize the cell membrane and allows the DNAs to enter. This is very important because, as you know, in bacteria, we don't have a nucleus and so a nuclear membrane. And if we destroy the entire cell membrane, the DNA is then exposed to the external environment. But we wanted to maintain a controlled system in order to allow the digestion of the nucleoid in our conditions, so by the DNAs that we use and not by other DNAs external of the bacterium. So we define this concentration as the optimal one for the entry of the DNAs and the cutting and recovery of the DNA in the S2 fraction. And then the S3 and S4 for the DNA region that can have interaction with protein or protein complexes or the condensed one. We perform this entire protocol on an exponential phase of growth of Helicobacter, and we reliably recovered distinct DNA fraction. And so we demonstrated that this protocol and technology can be used for prokaryotic cells and to study genome architecture. So what we did next was using this technology on different growth phases of Helicobacter, so also the stationary coccoid state and the biofilm structure in triplicates. Here you see some results that were performed by our collaborator from the CNR in Milan, so bioinformatic analysis, that are a little bit difficult, but what I wanted to show is that on the left here, we have a code of compartment. Is a kind of analysis that is usually due for ATAC-seq or high-C experiment in eukaryotic cells, in which we can identify an A and B state of the DNA. For eukaryotic cells, these are directly correlated with euchromatin and heterochromatin. So we can correlate the genomic position of the sequences to the A or B state. For prokaryotic cells, we cannot directly make the assumption that A and B are more or less condensed part of the genome, but we can only say that we were able to define two different states of the nucleoid, and that the genomic beams associated with one or another changed during the transition to the different state of Helicobacter. And specifically, 10% of these genomic beams shifted in the coccoid cells and biofilm structure. These shifting were not random because they were reproducible in all the different experiment, so indicating that there is a changing in genomic structure in the coccoid state that maybe is associated with a changing in genetic expression. Here on the right, I just wanted to report the sequencing profile of the S2 fraction that I remember you is the most accessible part of the nucleoid, and the S4, the most inaccessible one, in exponential and coccoid cells. Why? Because you can see differences in these two fractions and in these two state of the cells also at the macroscopical level. Because, for example, we have a loss of this peak in the S2 fraction when we are in the coccoid cells, but we have a more defined and separated peaks in the S4 fraction. So the DNA is more represented in the S4 fraction than in the others, indicating that maybe in coccoid cells we have a more compacted nucleoid of Helicobacter. In any case, these are preliminary analysis. Now they are trying to study the function of the different DNA fraction that we obtain in order to understand the biology under them and try to identify the mechanism and the genes that can change their state and expression during specifically the coccoid transition. So in the end, just the take-home message of this project is that, yes, H. pylori nucleoid is structurally organized and dynamic because it changes across physiological states and can be associated with a different gene expression during specifically the coccoid state. So in the general conclusion of this talk is that we can study the molecular biology processes of bacteria with different approaches. On one side, I show you how the ChIP-seq can help us define the regulon of a transcription regulator in Campylobacter jejuni, and that this protein is also sensible to oxidative stress. While on the other side, we develop a novel technology to study the nucleoid structure. So it is important also to remember that also the nucleoid architecture is another level of regulation of gene transcription in bacteria. So the message is that also these together, genome structure and transcription regulation, are interconnected and they can shape the bacterial adaptability, persistence, and response to environmental stresses. The future perspectives of this project are to integrate all these results with also other multi-omic approaches, for example, RNA-seq, but also RSEMI, in which we can recover the chromatin embedded RNA in the different fractions in order to understand if these regions are actively transcribed or not. Then try to investigate better the function of other factors, for example, HPTM43 in the nucleoid organization. And also we can translate all the experimental approaches that I show you on Campylobacter jejuni, but also in other bacteria to better understand the physiological processes. And these are all important and can pave the way to help identify new therapeutic strategies. So I conclude here. Thank you for your attention, and I'm open to questions.
Yes, thank you, ma'am.
Thank you, Dr. Elena. It was a nice presentation, which actually now I am thinking that I should actually start working on bacterias. Uh, so, so just a question from my side is that how do you look your technology role in using it in a cancer therapy or in a for cancer type of research work?
This therapy can be used to study, for example, how the transcription regulation or gene expression can change during the interaction, for example, between bacteria and cells, and see how which genes and how the genetic expression in general can be correlated with the pathogenic processes, and so the infection of the bacterium in cancer cells, and this can maybe help identify all the processes that are important for this bacterium in generating and inducing cancer in a human host. So I think that try to develop new techniques that can deepen the knowledge of physiological processes that can also be translated in all the clinical part of the research, and also help in this way, let's say.
Okay. Thanks. It was nice to hear from you. So you are basically now in Germany, right?
And yeah, just for a weekend, and then I will go
Okay.
back to Italy.
Okay. So the work which you have done is from Italy, from the same university where Enrico is there.
Yes, yes.
Okay.
I studied in Bologna. I had my PhD there. Yeah.
Okay. Thanks. Thank you. It was nice to hear from you. Thank you. Thank you
Thank you.
for actually accepting the invite and joining us for this fourth annual Swalife conference. Every year we do this, and major of the time Enrico is always there. So this time he referred you. Yeah.
Yeah.
Thank you.
Thank you
Thanks.
very much.
Thank you.
Thank you very much, ma'am, for delivering such an informative lecture. Your presentation has enriched our understanding of chromatin biology and regulatory mechanism in microbial research. We sincerely appreciate your valuable contribution to our conference. Thank you so much, ma'am.
Thank you.
Now we have reached one of the most awaited moments of the conference, the award ceremony. To contribute the award announcement, I respectfully invite Dr. Praveen Bode sir to kindly come on stage. Sir, may I request you to come over and proceed the award announcements. Okay, we will start our award ceremony at 3 p.m.
So, good afternoon, all, and welcome back to this second day of our fourth annual. Actually, this is the fourth year we are doing this conference every year in July, August. We actually do this conference. The purpose is to actually bring different interdisciplinary speakers who can actually contribute, who can actually share their research, share their the work which they are doing. But simultaneously, we wanted to make sure that the students who are doing UG, PG, PhD, and also the faculty who actually want to, share their research and that from their college or university without actually traveling. So that was the belief we started this conference, annual meeting conference for Swalife Biotech, and now we actually change it in different ways. It is not just now the annual conference, but we also have now uh, different area-wise conferences. So we had one in April where we were discussing about AI in drug discovery and development. We had speakers from Europe and UK. And simultaneously, we also have some other conferences now which will be there in October. That will be for innovation, entrepreneurship, and innovation. So I like to thank first now to the speakers who presented the posters and the oral presentation. Simultaneously, the evaluators who were there for last 2 days, the keynote speakers, and my team who actually handled all this for last 2 days. So now I'm going to talk about the result. So this year, actually, we started, Savitribai Phule Faculty Researcher Award, which I will be talking about it later. Before moving towards the, that faculty award, I want to first, announce the awards for the posters and the oral presentation. So total in these 2 days, total there were four keynote speakers, two yesterday and two today, and we had about 20 oral presentation, eight poster presentation, and those were from different category. We didn't have any actually UG category. We had PG, PhD scholars and faculty, and I really think that the work they have showed in their um, in their uh, presentation, that is oral or poster, it does reflect that there is major work is going on, which is helpful, and it will definitely bring some fruitful effect for Indian researcher community. So I will start with the conference oral presentation. So the first category is in the PhD scholar category for the poster, and that goes to Mr. Germany Dansana. He is the PhD research scholar. Then we have got second poster presentation award for faculty category. That is Mr. Sarang Mahamuni. He is assistant professor working in Krishna College of Pharmacy, Karad. Now I move to the next category, that is the oral presentation. And in oral presentation, we had PG, we had faculty, we had PhD scholar. So in PG category, the winner is Shrina Jha. She is a master student in pharmaceutics from SRM University. Then we have got oral presentation PhD scholar category. The winner is Rupali Nath. She is PhD research scholar, and she did actually showed her oral presentation, which was very um, effective work which she is doing. And in the faculty, we got Dr. Mona Lisa Chaudhary. She is assistant professor, and the work she has done was majorly on the in vitro studies on breast cancer cell line. So definitely, we had many speakers, and it was really difficult, to get through all these. People who actually has done the poster and the oral presentation, because it was very competitive. We had 20 oral presentation, and in that 20 oral presentation, maximum were from faculty and PhD scholars. We also had some presentations from oral presentations from dental doctors, and also some from the ophthalmic doctors. So it definitely actually showed that it was interdisciplinary, and I congrats all the winners of poster and the oral presentation. So thank you all for contributing and also showing your research. So now we will move to the next category. I will not say category, the award which we started. Now there is a purpose actually behind starting this award, and as a founder of Swalife Biotech, Swalife Foundation, and also the SSTSI community, I want to actually pass this message that why actually we started this award. Definitely, um, it does say that it is for the for the faculty who does research, but simultaneously our aim behind is that we want to make sure that this initiative is mainly taken for the encouraging and recognition of the faculty who actually go beyond the only the taking the teaching, but they go beyond and contribute in research, innovation, knowledge creation, and societal development. Now when I came to this idea about bringing this award, majorly the contribution was my role in UK as a PhD and master student. When I was in UK for 10 years, I have seen that the major research happen in academic, and when there is appreciation given to the faculty, definitely there is a research happening, the innovation is happening. And when I saw in India, in the academic area, that this is actually lacking, we decided that we will be having different categories in the awards for the faculty who is doing research. Simultaneously, there will be different categories of conferences where we will be also starting different awards in entrepreneurship and the startup. Now, majorly, if you look, the inspiration is the legacy of Kranti Jyoti Savitribai Phule, who actually demonstrated the transformative power of the education, and that's why we are celebrating that by actually bringing this Savitribai Phule Best Faculty Researcher Award for the faculty who actually do research, do mentorship to the student, innovation, and also the academic leadership. So the award aim to create a culture in which faculty members are not only teachers but also the innovators, mentors, researchers, and also knowledge creators who inspires the next generation of the student, and they can actually help in contributing a solution for different scientific, healthcare, and social challenges. So the long-term vision of this award is to actually recognize high quality and impactful faculty research, encourage faculty to move from teaching to research, innovation, and translation. So definitely the faculty who are following Swalife, Swalife Biotech, Swalife Foundation, and are in the community, they know that we are doing this for last 4 years, and awards we started this year, but we are doing different activities where we are promoting student activities, that is student mentorship, student publication, research culture, and we want to do it through the academician as with the help of the academic institute. Then third aim of this long-term vision is to recognize the contributors who are doing contribution in publication, patent, innovation, technology, and social impact. So that way we can encourage the interdisciplinary and emerging areas of research. So creating the visibility for promising researchers beyond conventional institutional boundary. We are trying to build a growing community of faculty, researcher, innovator, and research mentor. And that is why we are actually starting this faculty research award. Definitely, it was very hard. Actually, same what we had a problem with the I will not say problem, but difficulty with the conference to evaluate and bring the the winners. We have the same issues, same selection, difficulty with the faculty award as well. So to start with, actually the award was only um, uh, to actually when we decided this award, we had in the mind to actually give one award, and that was just from the nominees for just one faculty who is actually faculty and doing research. But then when we saw the nominees, there were 21 nominees from different faculty, different areas, and from different areas of India. We decided that we will have a runner-up, or we will have some sub categories as well. So I will start with that first three. We have got three runner-up here. So just to give you the first runner-up and the category as well. Professor Dr. Arsha Sultana, she played an important role in contributing in the research and the faculty. She is a professor in National Institute of Unani Medicine. She has got research going on into women health with authorship of 148 papers, leadership of about 18 funded research project, and supervised 47 doctoral postgraduate scholars. And she has also won three national awards from the government of India, and she is the runner-up of our lifetime achievement in medical and translational research. Definitely, her contribution is very very high in this space, and we really appreciate and thank you, Dr. Arshia Sultana, for what you are doing and nominating for our Savitribai Phule Best Faculty Research Award. Thank you. Then the next is Professor Dr. Nalini Shahu. We are actually having an award for him for his excellence in institutional leadership and research innovation. He is a dean and principal at Rama University, Kanpur. He has authored 122 research articles and 14 books, securing 20 patents and receiving the UP Garv Award in 2024, and also AICTE Azad Excellent Award. Thank you, sir. It is definitely an outstanding contribution, and we thank you for nominating and for this award as a excellence in institutional leadership and research innovation. Thank you. Then the third is Dr. B. Girirajan. He is Associate Professor and Dean of Innovation and Startup from SR University, Warangal. Um and the outstanding contribution he has done in intellectual properties and technology innovation. And for that purpose, we are giving him Outstanding Contributor Award, where he has got about 31 patents, six granted, which actually helps to increase his role in the technology innovation, driving student startup incubation as a dean of innovation and startup, and demonstrating leadership through the Ministry of Education and Educational Institutional Innovation Council, and his role as a innovation and startup ambassador, dean. He has contributed in the startup incubation center at the SR University. Thank you, sir, for nominating and playing important role in the innovation. Thank you. Now I will announce the winner of this first Savitribai Phule Best Faculty Research Award of this first year. That is Dr. Kuntal Das. Um, he has been, winner of this Savitribai Phule Best Faculty Award. He is a professor and head of Department of Pharmacognosy and Phytochemistry in Sri Siddhaganga College of Pharmacy from Karnataka. The selection committee of Swalife Foundation and Swalife Society for Translational Science and Innovation takes immense pride in selecting our first Savitribai Phule Best Research Award awardee Dr. Kuntal Das in recognizing of his contribution to natural product chemistry, plant biotechnology, intellectual property generation, and academic leadership. With over 23 years of dedicated teaching and 18 years of pioneering research, Dr. Das actually embodies of Savitribai Phule through his relentless pursuit of knowledge, scientific innovation, and student mentorship. I would like to actually highlight his key merits. So he has authored 152 peer-reviewed publication, out of which there are 125 research papers, 27 review papers, and more than 2,328 citation with H index of 26. Then he got 21 patents, out of which 12 published Indian patent and 9 granted patents are there. He got 15 high impact academic textbooks and 9 book chapters in different indexings like Elsewhere and Springers. He had mentored 18 young farm student, then 35 UG project student, and guiding one PhD scholar, securing 9 lakhs in competitive research funding. He is a global distinction. He has a global distinction by having a fellow of the Royal Society of Chemistry, which is based in UK, and a recipient of CV Raman Award 2024. So I think that's where this is an outstanding contribution as a faculty having a research and contributing in the community. I like to congrats Dr. Kuntal Das sir for his role he has been playing. Thank you, sir, for nominating and winning this award. Thank you. So now, after the announcement, I like to now invite all the winners. That is, Dr. Das, and Dr. Nalini, Dr. Girirajan sir, and Dr. Arsha madam, if they are available to spare their some time and give some valuable information about their academic and research. I like to invite all the four of them. So I think I can see Dr. Nalini is here. So hello, Dr. Nalini. Can you hear us? Hello. Okay, so I think, okay, yeah, he is there. Hello, Dr. Nalini Shahu. Can you hear us? Can you hear me? Okay, Dr. Kuntal Das is also here. Hello, Dr. Kuntal Das. Are you there? Can you hear us? Can you hear me?
Hello.
Yeah.
Yeah, good
Hello,
afternoon,
sir.
sir.
Good afternoon.
Yes, sir.
Congrats, sir. You are our first winner of our Savitribai Phule Best Faculty Research awardee. And I would
Thank you,
like,
sir. Thank you, sir.
yeah, can you share some of your, research and faculty experience with the.
Uh, I am myself Dr. Kuntal Das from Sri Sibanga College of Pharmacy, Singapore. So I am having around 23 years of experience, and my research orientation is in phytochemistry and all the business and chemistry. So I am engaging with the various isolation of the plant constituents and the analysis of various plant constituents, and we are doing further various pharmacological activity. And my one good thing is I can collaborate with some agriculture as well as horticulture universities, and I can go for various paper research projects. And I have received many research awards and research project also from various organizations. And generally, I am very much involved in endangered plant culture, which is very important and very interesting also. So you can go for the mass cultivation with the endangered national plant. So this is my area of interest. Thank you, sir.
Thank you, sir. It was nice to meet you, and definitely
Thank
we
you, sir.
can actually, collaborate and do some further activities. Thank you.
Yes, sir. Definitely, sir. Definitely, sir.
Thank you.
Thank you, sir. Thank you.
Over to you, Ruchira.
Sorry, sir. Okay. So good afternoon, everyone. Uh, Now we will move further to vote of thanks. So, as we have come to the conclusion of Swalife Biotech fourth annual international conference 2026, it's my privilege to deliver the vote of thanks. Uh, First and foremost, I extend my heartfelt gratitude to our keynote speakers, Dr. Praveen Bade sir, Dr. Nilesh Narayan Prasad sir, Dr. Deepak Behra sir, and Dr. Elena Chitte ma'am, and our winners for sharing their exceptional knowledge, valuable experience, and inspiring perspective. Your lectures have made this conference intellectually enriching and highly impactful. I sincerely thank all the researchers, faculty members, students, poster presenters, and oral presenters who enthusiastic presentation transformed this conference into vibrant platform for scientific learning and collaboration. A special note of appreciation goes to our esteemed judges for their valuable evaluation, insightful feedback, and encouragement to all participants. My sincere thanks also go to the organization committee, advisory board, technical team, volunteers, and every individual who worked tirelessly behind the scenes to ensure the smooth execution. We are greatly grateful to every delegate and participant for your active involvement, thoughtful decision, and continued support. Before we conclude, I would like to briefly introduce the organization that made this conference possible. Swalife Biotech. Swalife Biotech is dedicated to advancing healthcare innovation through scientific research, artificial intelligence, predictive medicine, drug discovery, clinical research, and biotechnology. The organization aims to bridge the gap between research and real world healthcare solutions by empowering researchers, students, clinicians, and healthcare professionals. Second is Swalife Academy. Swalife Academy is the educational division of Swalife committed to providing highly quality training, certification programs, workshops, internships, and skill development opportunities in clinical research, pharmacovigilance, artificial intelligence, regulatory affairs, predictive medicine, and other emerging healthcare domains. The Academy strives to nurture the next generation of healthcare innovators. Next is Swalife Consultancy. Swalife Consultancy provides expert guidance and strategic support to researchers, academic institutions, startups, pharmaceutical companies, and healthcare organizations. Its serves include research project consultation, scientific writing, publication support, regulatory guidance, AI implementation, and innovation strategy. Next is Swalife Services. Swalife Services offers a wide range of professional scientific and healthcare services, including literature review, medical writing, systemic reviews, and data analysis, AI assisted research solutions, project execution, clinical research support, and customized scientific solutions for academy and industry. As we conclude this memorable scientific gathering, let us continue working towards innovation, collaboration, and excellence in healthcare and research. Thank you all for your participation, encouragement, and unwavering support. We wish you a very thank you.